quase. Está. Está.
Good morning. We hope we find you and your families well and safe. The Polytech Institute of Bragança and the Mountain Research Center welcome you to the first natural product application, health, cosmetic, and food online conference. The Mountain Research Center is one of the 5 research centers within the Polytech Institute of Bragança and is an R&D unit of excellence. CIMU conducts research on the Mediterranean mountain system, following an interdisciplinary strategy that goes from nature to products. In all these years, we have had the commitment of disseminating science around the world, creating solid and robust bonds and partnerships with both academy and industry. And we are always looking for more challenging collaborations. In this sense, the Mountain Research Center gathers different ways to keep involving in our mission, science dissemination, especially now during this difficult pandemic situation in which science dissemination has been extremely affected. Therefore, one of our responses was the creation of this first edition of the Natural Product Application Online Congress, which consists in the dissemination of research using natural products applied in 3 different areas: cosmetic, food, and health. At this point, the NPA team and myself, Lillian Barrows, Vice Coordinator of the Mountain Research Center, would like to thank you all for participating in this exciting project, which in less than 1 month has received more than 450 registrations from universities to important companies from different parts of the world, such as Algeria, Argentina, Brazil, France, Greece, Italy, Mexico, Netherlands, Poland, Russia, Serbia, Slovenia, Spain, Ukraine, and the US. The NPA Congress received and processed more than 200 communications, from which the scientific committee has selected the most appropriate for each type of communication, considering the limited time we have for the conference. All the submitted works were divided into 3 main categories: oral, pitch, and poster communications, which will join also the 9 keynote lectures and 1 invited oral communication, to which we would like to thank their availability and for accepting this invitation. Finally, before leaving you with Professor Orlando Rodrigues, the president of the Institute Polytech of Bragança, the NPA team would also like to thank our special guest speaker for the opening ceremony, Professor Isabel Ferreira, Secretary of State of Inland Improvement of the Portuguese government. Professor Isabel Ferreira does not need any introduction due to her vast experience in food technology and natural products application, which she has been recognized for 7 consecutive years as a highly cited researcher by Clarivate, due to her more than 700 published research articles. She was also coordinator of several national and international research projects, has several national and international patents, and was supervisor of tons of postdocs, PhDs, and BA students. She has also been editor of many well-recognized scientific journals and is also the former vice president of IPB and coordinator of the Mountain Research Center. Thank you, Professor Isabel Ferreira, for being part of this opening session, which I know this also is a thematic within your great expertise when you were a researcher. Once again, we would like to thank you all for attending this first online edition of our Congress, and we will now leave you with the President of IPB, Professor Orlando Rodrigues, which I would also like to thank for accepting this invitation to be part of our opening session. Professor Orlando Rodrigues, as we normally would say, the floor is yours. But in this case, the screen is yours.
Thank you very much, Lillian. Good morning, everyone, and thank you very much for coming to this online participated Congress. Just a word to thank you, all of you, to be engaged on that initiative. And a very special thanks to Isabel Ferreira, our Secretary of State for the interior cohesion. And thank you very much, all of you, for organizing this so important meeting. It's very interesting that you continue working in cooperation all over the world and in the field so important nowadays in our societies. Indeed, you are in the center of some very important priorities for our societies all over the world, not just in Europe or in some regions of the world, all over the world. The sustainability of the way we do our industry, our food production, it's very important nowadays. And your research is in the center of that. We need to take more value from agriculture and natural products. We need to do the production of food and in general industry production in a more sustainable way. There are a lot of changes that are now in our industry, in our production, and really your research is in the center of the most important things that we should do in these days. So thank you very much for doing what you do. Thank you very much for cooperation. It's very important that researchers are probably the people that more cooperate between countries all over the world. It's very important that you continue doing that. Of course, it will be very important that we can do this kind of sessions presentially, but it is also very important that we take profit from that way of working and being in contact with each other, and in the future we do more contacts, more cooperation in these digital ways. So thank you very much for being engaged, and a lot of success for your initiative. Thank you very much.
Thank you, Professor Orlando, for your kind words. And now I will share the screen with Professor Isabel Ferreira.
Thank you very much. First of all, I would like to present my regards to the president of the IPB and to the organizing committee and also the scientific committee of this very, very, very well participated meeting, and particularly in the person of Dr. Lilian Barros, which is the leader of an excellent and brilliant team that works on these topics of natural products. So I am very proud to be here and share the screen with all of you. And the research on natural products has been receiving huge attention in the last decades. And plants, mushrooms, and algae contain, as we all know, a wide diversity of bioactive compounds that may play an essential role in the development of health, cosmetic, or food products, which is the topic of this meeting. In what concerns food products, there's an overall debate regarding the safety aspects, namely of artificial preservatives added widely in many food formulations to prevent the multiplication of spoilage and pathogenic microbes. This is obviously an arguable issue since all additives employed in food products are priorly subjected to a strict evaluation and validation process conducted by entities such as Food and Drug Administration in the US, but also European Food Safety Authority in European Union. Therefore, their safety should be considered as indisputable. Nonetheless, as we all know, there are several reports, and mostly scientific reports, highlighting particularly deleterious effects evaluated in artificial compounds, such as carcinogenic or teratogenic attributes or some residual toxicity. This ends up, of course, setting pavement and intensive research oriented towards finding natural surrogates to replace the artificial or synthetic compounds that are usually associated to those adverse effects. Likewise, to prevent the aforementioned problems, consumers and authorities have been stressing out the need to substitute these artificial additives with alternative, effective, and non-toxic substances, and natural products could be an alternative. When added into a determined product, these natural agents might be employed to protect, but also to enrich the product itself to benefit the physiological state of the consumer. For example, natural antioxidants are well known for their wide range of biological and inclusively pharmacological activities, therefore representing several beneficial effects in nutrition and also in health. And for these, antioxidants contribute to increase the shelf life of a wide diversity of products, as they prevent the oxidation of several key compounds, which, if oxidized, would seriously compromise the product quality. Other agents, such as, for instance, antimicrobial compounds, are added to protect the product for unwanted microbial-based spoilage process. But when employed in food products, they should not exert any physiological antimicrobial activity. Independently of the targeted purpose, if we plan to extend the applicability of natural substances in health, cosmetic, or food-related products, it is mandatory to increase our knowledge on these versatile natural additives. In any given study, it will always be essential to perform molecular structure elucidation analysis, clarify the molecular cell mechanisms through which microorganisms respond against natural bioactive compounds, to elucidate all potential structure-activity relationships, to comprehend different matrix effects on the efficiency of biological activity in combination with other hurdles, use emerging technologies like microencapsulation techniques or others to increase the biological activity of different natural agents, and also to understand consumers' acceptability and their quality perception. And as I previously mentioned, intensive research has been conducted to identify and characterize this nearly endless diversity of biological compounds. This through work has originated an immense library of compounds and big data on their associated bioactivities. However, there is a lot to be done in the scientific community-industry interface. The Portuguese government is highly committed to strengthen the collaborative work among these two key actors, as clearly exemplified by collaborative laboratories, but also research center as the Mountain Research Center, which has an extensive work with companies and with the industrial sector. And the intensive activity in co-promotion projects. And for that, the government will continue to fortify the diversification and differentiation of experts ecosystems, technological centers, ID institutions, and industry, aiming to capacitate the introduction of novel processes and products, able to reach market success and to impact the economies of the territories. This integrated approach is entirely displayed in the next mobilizing agenda, whose expected promotion of technological transformation will be supported by the national clusters with highest potential in specific scientific sectors, as identified in our program, Expandir, the program of expansion nationwide projects for interior development. This was recently approved through a Minister Council resolution. And in what concerns to these matters, to the agro-food sector, we have clusters related also with the development of natural-based health, cosmetic, and food products. And they are located all over the country in different cities, including Bragança or Elvas or Castelo Branco, in all regions of Portugal. Depending on its specific application, natural-based products must comply with highly strict rules in order to be included in European markets. The majority of natural ingredients for the health sector is used in supplements and herbal medicinal products. And herbal medicinal products must be, of course, in compliance with several directives. The European Medicines Agency is the organism responsible for the scientific evaluation, supervision, and safety monitoring of medicines in member states. This commitment can make about herbal medicinal products as well as use ingredients. There are mandatory legal requirements for food supplements, particular in the European Union food supplement legislation and in the European General Food Law. And the new novel food regulation approved in January 2018 is another essential tool to ensuring food safety. Established ingredients for food supplements are listed in the Novel Food Catalog, the Union List, and the Botanical Lists for food supplements. In addition, there is the need to provide data also, of course, on toxicological, microbiological, and allergenic properties. So all in all, the potential of natural products is immense, and every effort should be dedicated to use this potential in differentiated industrial application, always complying with the strict legal framework that rules these particular industrial sectors. Thank you very much for this opportunity, and I wish you all a very good Congress. Thank you.
Thank you, Professor Isabel, for your words. They're always very inspiring, and they inspired me during all my life. So I thank you very much for your time to be with us in this opening session, which I also know that this topic is within a theme really close to you and in which you worked along many years. And for us, it's a great pleasure to have you here with us today. So thank you for your words.
Thank you. Goodbye.
Goodbye. Now I will pass the floor to our moderator for this section, This Congress is divided, like I mentioned, in 3 areas: health, food, and cosmetic. This morning, we will be talking about health, natural products linked to health. And the moderator today will be, for this section, will be Dr. Soraya Falcão. So I will give the screen to Soraya at this moment.
Good morning. Next, we'll have the presentation of 2 keynotes, each one with a duration of 25 minutes, followed by 5 minutes of questions. The public can ask the questions that arise in the chat of the YouTube channel. Some of the questions will be selected and will be asked to the speakers in the end of their presentations. Just a brief note about the posters. The posters are available online for consultation, and they are organized by category, and they can be downloaded. So, for the first keynote, I'll have the pleasure to present Professor Maria Graça Campos. Professor Maria Graça Campos is Professor of Pharmacognosy at the Faculty of Pharmacy, University of Coimbra, and she is coordinator of the Observatory of Drug-Herb Interactions. The main topics of research includes drug discovery, especially with byproducts, and pharmacovigilance in drug-herb interactions, mainly with oncologic patients. Her keynote is entitled Globalization of Beehive Products in Food and Health. So I pass the word to Professor Maria Graça Campos.
Soraya, Maria Graça Campos is 5 minutes delayed, or 2. I'm just waiting for her.
No, it's here. She's online.
She's online? Okay.
Yes.
Okay.
Just give me some minutes. Okay, we are all here. So, good morning to everyone. Do you want that I start now? I share my screen?
Yes, of course.
Okay. Okay, good morning to everyone. Before we go further, I want to thank the organizing committee the invitation to be here today to share with you some data, some thoughts, and some concerns, of course. I also want to thank to all the players involved with the products, of course, including to all my team and all the researchers that I work with. In these next minutes, I'm going to be taking a look at the product's potential based on the research that is already done. And I will show you some of these works around the world. So I will highlight what I see as the main areas of expertise. And to do that, I choose in a random way, as you imagine, some publications from different countries correlated to different bee products, just to give you a broad idea what is going on around the world. Of course, I could not put all. So forgive me if I didn't put some of your work. I imagine that some of you that are now assisting have done very good research. And for the others that are just beginning, I did file a zip file that I sent to the organizing committee with a good amount of very recent publications. Some are cited in this presentation, but others I didn't have the place to put them because, as I said, they were too much. So please feel free to ask to the organization this presentation that is also in this zip file, and of course all the publications that I put there. So let's get started. When we talk about globalization, of course, we talk about quality. Without that, we cannot move the products around the world, and we cannot have the same function for all of them. So it's fundamental that we have products that are standardized. Be sure about the storage, and of course, to add value to these products, we need innovation. I will talk briefly about some applications of bee products in food and pharmaceutical industry. We have now a good amount of information that is collected for I could say more than 100 scientists, colleagues that work in different levels with the bee products, and the standard methods to work with these products are already published in Colos bee book, and the missing ones, pollen in venom, will be published this year. So for standard methods of analysis, we have already a good tool where we can go get this information. Just for highlight some of the products they are already categorizing as food or as pharmaceuticals. I show you here these Codex Alimentaries that you know. And there we have already a standard method for honey. So it's already classified as a food. However, I hope that in the near future, we could have also bee pollen and bee bread categorizing as a food too, because they have all the potential. What we need is, in fact, do what is already completed. I will show in the next slide. For pharmaceutical purposes, we have also our Holy Bible. So our Holy Bible are the pharmacopoeias around the world. Soon we will have a global pharmacopoeia. We are working on this in World Health Organization. But for now, for example, I show you what we have in the European one. And there we have already 3 products from the hive. As you can see, we have bee wax and we have honey. Those are the monographs for quality control of these products to be used in pharmacy. And we have also EPIs themselves. So we use them as in homeopathic preparations. And as you can see, we have already hive products introduced in pharmacopoeias, and I hope that all the others could be very soon introduced too for pharmaceutical purposes, because as I will show later on in this presentation, we have a huge potential of these products in this field. In many ways, we are working also in ISO normalization. You know that these norms are not mandatory, Nevertheless, they are fundamental to normalize some of the information that should be done with these products. We have now these working groups. We are, Ophelia and myself, we are in Bipollen group. We know that in most of them we have Portuguese people, Portuguese researchers. Thank you all for the nice work that you are doing. And for Poland, we have Ophelia as convener and Wang Liang as secretary. The other groups are more advanced in the norms, so I hope that we can catch them. Well, let's move for foods industry. We have products that are clearly involved in food or as a food because they have an important nutritional content. Otherwise, we can also use them in processing due to these important bioactivities. So I will show some of the examples that we have in a different way that we can use these products. For example, for bee bread now we are trying to get the best information possible to classify the nutritional content selected by a species, what was at the beginning something that probably we didn't think that we could do. But in this work that we published with colleagues in Romania, we were able to identify clearly the species, the origin species. So this was one sample of Brassica and another one from eucalyptus in different regions in Romania, what means that we can go farther in the use of the product if we classify the floral origin and we'll have a reproducibility of the product. For pollen, we know almost everything that we could know about pollen. Research in pollen is already, I could say almost complete because we know so many things about the nutrients, the micronutrients. Sorry, I missed here the vitamins. I'm really, really sorry. And we know lots of bioactivities as I will show in the next minute. With pollen, we know already clearly very good amount of floral origins. They are distributed around the world. And for all of them, as I will show, we can get important bioactivities and clearly application in industry. For example, we have this work that we did with Ethiopia. We have here coffee and tobacco pollens. They are very, very specific. And here we have coconut pollen. And Brazil, for instance, have tons of these products. And some of the research carried out is anti-tumor. So we have important application of these products in the industry. Moving to processing, when we want to get the best of a product, we need to get the best of the bioactivities, and the storage is fundamental to be sure that we get at the end the same bioactivity that we have at the beginning. These colleagues from Bragança and from Castelo Branco, they did a very important research exactly showing the importance of frozen bee pollen to get the best prevention of oxidation for lipids, for instance. And this allowed to use this bioactivity in innovation. And for innovation, what they did was compare the conventional antioxidant with the pollen itself and the extract. They are black puddings and very typical in Portugal. And they verify that these two products could prevent better than the conventional antioxidant. They verified that bee pollen was a little bit more active than the extract. This happened many times. However, depending what we want to do in industry, in my point of view, sometimes it's better to use the extract because they are more easily to standardize. And I think it's a very good application for some of the compounds in bee pollen. Yanka and our group also did a similar essay, but with cookies. In this case, we had, as in the other one, of course, we had compounds, and these compounds can fortify the products. What is quite interesting, when she was working with us, we did a work with beer. It's not published yet, but probably in the next conference, I can show you this data. So as I said, we have other examples in the bibliography, and we can see them in this zip file where I put some of these papers. Well, going forward, I think everyone knows honey. What can I say that you don't know already? It's clear that we have a high content of sugar, but we also have some micronutrients. I leave here just a paper from Morocco, just to show you that, as I said, I choose different countries around the world. We have lots of information about honeys from different countries. And this is really good because each of these countries can explore the flora that they have. And coming back, what I was saying is, for example, a good application for honey is in dehydrated people. So you can dilute the honey and give in the water because will help even to restore the intestines microorganisms. Well, now my field, because as you know, I work in pharmacy, so this is more my area. For all these products, I will base the next slides of this presentation in this chapter that I wrote with Ophelia and Helmedy for the book No Bees, No Life. And also in these two papers, this one I wrote with the colleagues from China and Turkey in Italy about royal jelly. And for bee pollen and bee bread, it's a joint venture with Romania and Turkey. Well, I must say that I use the materials that we have, that we prepared to these papers. So some of the tables look smaller. I didn't want to cut them too much, but I highlight what are the main points to think about. And you also have these papers in the chapter in the folder, so please feel free to get them. Well, for walks, we'll start with walks. Walks, as I show you at the beginning, is already in the pharmacopoeias, not only in the European pharmacopoeia, but in many others. What we have here are mainly glycerides, fatty acids, and long-chain alcohols. So it's easy to understand that this mix gives a product with a very good stability, didn't vary with pH or moisture levels. It's wonderful to be used in pharmaceutical technology due to the hydrophobic matrices that can be used in drug delivery, in tablets, suspensions, implants, and microspheres. So a huge potential in pharmacy and in fact can add value to the product. Moving to royal jelly, we did a comparison among the main compounds in different products of Beehive, but what is important here is that once again we need to have clear information about the species, the floral origin of the product. This will change a little bit in the amounts and kind of components that we'll have inside. In this scheme, we can see clearly how we do drug discovery. So we start the main matrix, we check the compounds inside, we split in different isolated compounds, and after yes, we can go to the bioactivities. So we have here antimicrobial, home healing, some information about the possibility to use as anti-cancer, what are the compounds that we can use for. And this is the way to go. Otherwise, the results are not reproducible. We can have always the same thing with the same product if we don't have this information rationalized and well categorized. So the main bioactivities for royal jelly, as we classify for the information that we have more, are antioxidant activity, wound healing, immunomodulatory, and anti-aging. As you can see here, we also know already what are the main mechanisms of action, and this allowed us to go further in development of a new drug, because otherwise we could not establish the efficacy, the toxicity. So if these compounds are not safe, we cannot use them for drug discovery. Sorry, I need to take some water. Well, as I said, I think most of us know lots of things about honey, but I could not moving forward without talking about the work of Peter Mullen. Peter Mullen is a professor from New Zealand. I had the opportunity to work with him when I was there. And he was the father of the application of honey in wound healing, with all the information that allowed us now to have in the hospitals, the bandage that we can use in burns, in surgical wounds. And in fact, it was a good step and an important step in the evolution of this product. Of course, all this research was carried out with Metacia, endemic from New Zealand, Manuka. But for example, for phenols and flavonoids in Poland, the compounds are very, very similar to our eucalyptus. I think that maybe someone could do similar research here. Well, another application that usually we attribute to Propolis is the antiviral activity. But we also have good data for the same use with honey, in this case in herbs. For propolis, please be careful with the preparation of the extract, because some of the compounds can in fact induce allergies in skin. So before apply in the lips, anything that could cause allergy or even give some color to the skin should be taken out. Well, I work in Poland in bee bread, as you already have understand, I think. So I will detail a little bit more this data. We did in this paper some tables, long tables, by the way, but I just choose some of the information from them about the main bioactivities that we have a good correlation among the original floral origin and the bioactivities. You have here some examples for the anticarcinogenic bioactivity, brassica, cistus, salix, anti-inflammatory also, and for cistus, in fact, we have good data about the activity in osteoporosis. You know that this is a kind of problem that nowadays it's important to help in this therapeutic pathology. Well, with this pathology with other tools in therapy. So it's very important if we could develop a drug using these extracts. For the hepatoprotective activity, the data that we get are from Castania and Brassica pollen. However, I must alert that all the hepatoprotective products that we have, didn't matter if they are these ones or extracts from plants, or even chemicals prepared just by synthesis, they are products that when they act in the liver, they can produce drug interactions. So if people are taking drugs, please avoid this kind of product. Otherwise, something can go wrong in the therapeutic. Well, moving on in these bioactivities for bee pollen and bee bread, we also found a huge activity antimicrobial. I think that is a kind of bioactivity that is crossing all the products in beehive. And we have lots of data for these different microorganisms and also for huge variety of floral origins. In Portugal, we have a nice group working on this. So they also get lots of information from different species and with different microorganisms that we can use honey, honey, of course, and in this case, bee pollen. Another bioactivity that is quite explored in lots of products and also in bee pollen is the antioxidant bioactivity. In the most of the cases, this bioactivity is due to flavonoids or phenolics in general. What is important is that nowadays we have an increase in degenerative pathologies, Alzheimer, Huntington's, Parkinson, arthritis, and the possibility to use compounds with antioxidant power, it will be possible a way to go. So recently we wrote this paper for the Huntington's disease. This is a genetic mutation that induces this disease. So we don't have a cure for this pathology, but we can manage to delay the symptoms of the disease. And for the other pathologies that I showed before, Alzheimer, Parkinson, arthritis, probably we could use the same way to go. So here we discuss the application, the use of dietary flavonoids, in fact, to help the patients with this pathology. And of course, as I said, we can apply for others. Another new application is the use of bee pollen, and in this case, in a mix with propolis, to reduce neuroinflammation in a model of autism. Here they use the effect of a prebiotic rich diet in this model to decrease neuroinflammation. However, please take care when we extrapolate this information for humans, because the use of prebiotics, probiotics, or even the symbiotics when we put all together is not yet well proven. So we know exactly if they stay in bowels, in the intestines, and they can exert the effect that theoretical we think that they do. Now, the last bioactivity is antitumoral. As you know, we have also an increase in oncology. We have lots of patients and new, better and more antitumoral drugs are in fact needed. So some of the information that is now carried out with this product and even with the others that I will show are fundamental to achieve new drugs. We have already some clinical trials going on, but we still need to go and to do lots and lots of much more to complete the data that is needed to can have these new drugs. We have other products from behind under this investigation, as for example, Propolis. Our colleagues from Bragança are also doing a wonderful job on this issue. And another product that is explored with this bioactivity is epitoxin. Mainly at the beginning, we use it for relief pain or chronic inflammatory diseases, but we have already information in some papers that we can explore the antitumoral activity with success. Helmedi, with the colleagues from Bragança, are also in his PhD degree exploring these potentials. So as you can see from these hive products, we can have a possibility to develop important drugs for health application. So just recapping, importance to have a floral origin or the identification of the product. Standard methods, I think we have already a good work done. Normalization, we are going, we are completing the data sooner, I think. And of course, with all this, if we validate the bioactivities, we will have an important add value to this product. Thank you so much.
Thank you, Professor Maria Graça Campos, for this excellent review on the byproducts. So now, before going to the questions, Professor Graça shared with the Congress organization all the material used in the presentation. If someone wants to consult, just send an email to the Congress organization. So now we'll pass to the questions. So I'm going to ask. I don't know if we have questions from the public. I think no, but I'm going to ask some. You clarified the potentials and the handicaps in the use of the byproducts. But in your opinion, what are the main handicaps, or which steps we have to overcome to pass from the studies in the laboratory to the use of the consumers in the health area, for example?
We need to clarify. So I cannot use, for example, the pollen directly or propolis or epitoxin in oncology, of course. So for health, if I want to use in oncology these products, it's clear that we need to isolate the main compounds that are responsible for this bioactivity, and we need to carry all the steps. So finish all we have already done and go to the clinical trials, and after, of course, we can develop a drug. Otherwise, we have, for example, the possibility to use honey to hydrate people, as I said at the beginning. In some cases, in nutritional point of view, we can have these products as bee bread or bee pollen because they have a high nutritional content. However, for homemade is what you want to say. We can use these products homemade just in our home, or just going to the industry, because they are two different things.
Yes, I know, but it's things, we see a lot of scientific works telling this propolis is good for this, bee pollen is good for this. So there's always a big handicap between the scientific, the information that goes out from a lab and then to the consumers, the use of the consumers.
Well, I think that in this century we should move on with all the data that we have from research. We should move on from the traditional medicine and validated these products with all the safety information that we have. Imagine, for example, what I said for propolis that we can use for antimicrobial activity. In fact, and you work on that, but if I have an infection, I will not take propolis, I will take antibiotic. That is already prepared in the right formulation. So I know that is the efficacy and the safety for these doses that I need to take to fight this infection. So for me, it's quite difficult to use. I know, and I apply a few things in my family, let's say, and I also give lectures in phytotherapy. So I teach how to use some medicinal products, but we need to be very, very careful with the side effects and the toxicity. And for example, propolis is very good, but it's also very toxic. As for example, epitoxin, okay? It's differently if I talk about honey or if I talk about pollen. Those are clearly products that I can use with a nutritional value. And in certain cases, I can use them to give a little bit more of bioactivity. It's the difference. If I wanted to, so the big idea is we have a homeostasis in our body that should be put in certain levels, okay? If I have a small imbalance on this homeostasis, I can use some of these products. But if the homeostasis have huge differences in labs, I need to use drugs, proper drugs that I have the right dose and I know all about the side effects. And even in these cases, I can have collateral effects that are not very good. My concern is more, imagine that we use one of these products and we have an important side effect. This will kill the product. This goes in the news and will kill the product. So we need to be very careful with the advice that we give to population to take these natural products just to cure without a good reflection.
So we have a question from the public.
Okay.
Are there standardized tests for the antibacterial properties of honey and for wound healing?
Wound healing.
The first is antibacterial properties.
Okay, we don't have these, I think, in Colos. Anyway, well, yes, you have. We have for pollen, not for honey, not for honey. In the Colospi book, and I think is free for consultation in the web. You can go for these chapters. I don't know if they have for honey, but we have for bee pollen. By the way, this part of our chapter was written by Leticia and some of their collaborators. So yes, we have there what we propose as a standard method. So what we did was looking in all the methods that were already published and select what could be the best one to be used by all the researchers. Yes.
Okay.
Healing. I don't remember to find anything.
Okay, so we don't have more questions. Again, I thank you for the excellent presentation and thank you for being present here in this Congress. Thank you.
Thank you too. Thank you. Good luck.
Yes, now we'll pass to the second keynote, which is my pleasure to present Dr. Pedro Cantista. He is a medical doctor specialized in physical and rehabilitation medicine, competencies in medical hydrology and pain medicine, consultant at the Centro Hospitalar Universitário do Porto, professor at the University of Oporto, president of the International Society of Medical Hydrology and of the Portuguese Society of Medical Hydrology and of the Portuguese Society, I'm repeating, of the medical hydrology, sorry. So the keynote will be entitled Thermal Products, Mineral Water, Pelloids and Gases.
Thank you very much for your introducing words, Dr. Soraya Falcão. Before I start my presentation, I would like to salute every one of you, dear participants, and also to thank the kind invitation to be present here to the Mountain Research Center of the Bragança Polytech Institute, especially to Professor Maria José Alves and Dr. Lilian Barros. Thank you very much for your assistance during the preparation of this conference. And I would like to address my best regards to Professor Isabel Ferreira, Secretary of State of our government for the interior cohesion of our country, of Portugal, and also to Professor Orlando Rodrigues, President of the Bragança Polytech Institute. And also to thank the excellent keynote lecture of Professor Maria Graça Campos. I learned a lot about bees and bee products, and I am very interested, I must say, because I dedicate also some of my research to osteoporosis, and I will contact you about the mechanisms of these products in osteoporosis. So this is a challenge. I stay here. So let me share with you my screen now. I hope that I can enter this. So let me see. Let me see if I can now. Just a moment. What happens now with this? Not here. Are you, I think it's now okay. Is it okay?
Yes.
Yes, okay. So let's start. So I was asked to speak about this topic, thermal products. And as you may imagine, I only can make a very synthetic approach to this subject due to the short time we have. Nevertheless, I hope this presentation may be useful for you and give some clues to develop your study and motivate your interest and knowledge about thermalism. Well, what are thermal products? By thermal products, I mean the substances used for treatments within thermal medicine. And these include especially mineral waters, fluids, and thermal gases. And I must say that these products, special mineral waters and fluids, are used since thousands of years in almost all human civilizations. Of course, initially, this use was purely empiric practice, but nowadays it has a scientific basis and may be included in what we call evidence-based medicines. This means the evidence by the perform of randomized controlled trials, which is considered the level 1A of evidence in EBM. Well, despite the fact that we have already acquired a lot of scientific knowledge, research is of course much needed on this field. The first idea and the first thought that I would like to share with you is about the need to have an international glossary in this field. In fact, the first step to develop cooperation and exchange of knowledge on scientific research is to speak the same language. This means that we should respect an international glossary. And of course, why that? Because nowadays, words are not only to speak, but they serve as keys and codes to have access to automatic information, informatics, and to spread it into the scientific knowledge to obtain an international, a global cooperation. So we need to use the same terms corresponding to the same concepts and definitions if we want to reach and spread the right information. I give you an example. Usually you talk about thermal water, but in fact, the consensualized term nowadays is no more thermal. Instead, we use mineral water or natural mineral water. So temperature is no more the defining criteria for define this kind of natural product. In fact, when we talked before about thermal, we referred about water that came in a source with a temperature above the body's temperature, which means above 36, 37 grades Celsius. Then it changed for the concept, and we refer to a water that emerged in a temperature above the ambient average. For instance, 26, 27 if the local average during the year would be, for instance, 21 or 22. So nowadays, the criteria to define a natural mineral water is the therapeutic effect. So as you see, when I talk about thermal water, if I publish about thermal water, I can miss the target if I don't use as keyword or mesh word natural mineral water. Nevertheless, this need, there isn't yet an universal consensus, since low mineralized water, for instance, is not considered mineral water in many countries. If you go to, for instance, for East Europe, they don't consider a low mineralized water like our Água de Luz in Portugal, like Luz water, because it has a low mineral content. But in fact, if we take as criteria for the definition the therapeutic effect, the fact is that we have already some evidence of the pharmacologic effects, let's say, that with this low mineralized water. So our International Society of Medical Hydrology, which, by the way, this year accomplished 100 years, it was founded in 1921, adopt as glossary these two books. The main one is on your right side of the screen, was written by Professor Maria de Los Angeles Ceballos Hernández from Universidad Complutense de Madrid, and it puts the equivalent terms in several languages. It is very complete, and I think that is a wonderful tool for those who want to publish and study on this field. We have a second glossary, but only in German and English by Christoph Kirchner. Anyway, I think that they are very useful for you to, for those who are interested in communicating in these fields. The other publication of reference, and which is now the adopt criteria for definition and concepts, is that one published in 2010 and adopted in that precise year at the Congress in Paris as the definitions validated by the International Society. So this is a paper in which I have also the honor to participate by Gutenbrunner, Bender, Karagul, and myself. The other product that we're going to approach here is Pelowitz. Now, by Pelowitz is the umbrella designation for many natural products, and they were classified and systematized since 1949 in our International Society conference in Dax, France, near Bordeaux. And here you have the publication that currently is the reference for this international consensus, published by Professor Celso Gomes from the Aveiro University and more 9 collaborators trying to have let's say, share the consensus especially in Europe. So you have here the reference of the publication, and I can send it's a free access publication on the web. But as you see, this is the classification, the table of the classification in Dax in France. It is in French, because at that time, French was also a very used scientific language. Nowadays, we don't use French. Unfortunately, in my point of view, we only use English. 200 years ago, we used Latin, so we need that equivalence. That's why we need an international glossary to have the same definitions in different language corresponding to the same concept. And that's why we need that correspondence in English. And in some fields, I must say that English is not enough. English is a very easy language with a very simple grammatics, but sometimes, the vocabulary, the terms miss, and we need to go another way to Greek or Latin to incorporate these Latin and Greek terms in English and have the correspondent equivalents. So here you have the denomination, you have the origin, you have the chemical nature, you have the temperature, you have the conditions of maturations, and you see boue in French, fungi, Italian, muds in English, schlam, German, and then all the designations, and sometimes you see mud, peat, fungi, and in fact, all these designations correspond to different kinds of pelloids. So again, if you are interested in developing the research in these very interesting natural products with the pharmacologic effects and therapeutic effects, very interesting, you need to choose the correct words to define what you are talking about. So mainly and very synthetically, I would say that these peloids effects may be anti-inflammatory, quite powerful, I must say. There are trials that compare, for instance, the anti-inflammatory power of some peloids in Italy, for instance, with indomethacin and also with diclofenac, and they are equivalent in power. This is really amazing. I myself, I must confess, I was amazed when I saw that work, especially from Professor Simona Bellometti. Okay, you have also other effects, namely on your skin, keratolytic, of course, according to the kind of composition of the specific agent. Speaking about natural gases, the main are these three: CO2, sulfur gas, H2S, and radon. Of course, when we talk about radon, we're talking about very small amounts of radon, where we exclude, of course, products that have high levels of radioactivity. So these are some of the CO2 effects. They are quite important, for instance, on acting on the peripheral vascular resistance, mainly on hypertensive conditions. For instance, also on arrhythmias. So you have the capacity of influence heart rate variability. Also in skin and localized adiposities. And as a narcotic therapy could be an important method for treatment of some kinds of neuroses and psychosomatic conditions. Concerning sulfur gas, I would say that it can mediate regulation of apoptosis, signaling ameliorates adverse cardiac remodeling and diabetic cardiomyopathy. It gives some myocardial protection against ischemia and reperfusion injury. It also seems to have therapeutic benefits on Alzheimer's disease, and very recently some studies pointed H2S as a potential defense against COVID-19. Of course, this research is in the beginning, but there are some interesting findings about this. Concerning radon therapy, well, I would say that radon has mostly an anti-inflammatory effect, and there are several methods of administrating radon in water baths. This is very old, since, for instance, 1996 in Czech Republic or in Bad Gastein in Austria. Also by drinking therapy, which you call hydropenia, for instance, in Bad Brambach in Germany, inhalation in Poland, in the United States, in Austria also. And in the United States, in Europe, there are several radon spas specialized on radon therapy with high radon atmosphere, and also in health mines established in Bastion and Montana. I'm not at all an expert on radon therapy, but our colleague Albert Falkenbach published a lot about this and has interesting findings about this kind of therapy. Now, I will finish by, I think, the main product that we use in Europe regarding spa therapy products, which are mineral waters. And the current definition of mineral waters is natural waters, of course, of deep circulation. This is very important. It's not lakes, it's not rivers. We're calling about water that infiltrates the deep, as a long way in subterranean geologic formations. And during that long circulation, it happens that it gains some soluble contents, some mineral contents, also some biological contents. So the physical and chemical characteristics allows therapeutics properties to be attributed to them. Mineral waters has a very stable composition, so if you go to a source, each mineral water has her own identity, and it practically don't change along all the year. So we're talking about a period of circulation underground, like, for instance, 9th centuries, 10th centuries. So if you drink, for instance, Água das Pedras from our region, you are drinking a water that came from the rain almost 1,000 years ago. So this is very important. That's why that long way gives the possibility of getting the stability. I must say that on the designation of mineral waters, we can also include the gaseous waters, even if the gas content could be reinforced with the gas from the source itself. It is the only change that is allowed to do in a mineral water. Why mineral waters acts? Because they have several therapeutic factors, mechanisms. Now, physical mechanisms, it is different to take a bath on, for instance, on a tube with a water of 34 degrees or 38. It is different according to the chemical composition. And I must say that some biological contents are also useful to have some therapeutic properties. Some vestiges of, for instance, RNA can give some properties to the water and give also the identity. Well, this is not yet still studied, but is a very important research line, for instance, here in Portugal. And of course, you know that you cannot deny the very important psychological effects, and the psychological effects indirectly influence your, let's say, interaction with the waters by, for instance, immunomodulation. I deeply believe on this. Okay, so you have a multitude of mineral waters. You have thousands of mineral waters in our nation, so you need to know them better, to systemize, to classify it. And usually we use 5 criteria for classified water, to classify water: physical and biological and chemical. Now, the temperature, the osmotic pressure, radioactivity, mineralization, and chemical composition. By the way, I let you hear a curiosity. One of the oldest treatises of classification of mineral waters from a very well-known doctor from the 18th century, Dr. Mirandela, because he was from our region, from the city of Mirandela, Francisco da Fonseca Henriques. He was a doctor on the court of our king João V, John V. And this was published in 1726. It was very complete, and it is still very, let's say, very, very actual here, very, very current. This was considered the first compendium book of classification of systemization of the Portuguese mineral waters. Here you have. Recently, it was republished under a facsimile edition. I will talk only about the criteria of mineralization and chemical composition because we don't have time to approach more topics. I dare say that there is not a uniform accept criteria for these thresholds on mineralization. It varies from school to school, even in Portugal, from the school of Coimbra to Lisbon, and of course from country to country. Nevertheless, I think that nowadays the international society is reaching an international consensus about this, and I hope that at least in the next 2 or 3 years we will have it definitely published. Now, I give you some examples here. I don't have time to enter into details, but anyway, you can see that we have waters weakly mineralized, low mineralized, strongly mineralized, and here you have the difference of the contents, for instance, from very weakly mineralized, like again, Luz or Vitalis in Portugal, you have less than 50 milligrams per liter. And then if you go, for instance, to a high mineralized, you can have 2 grams, for instance, 2000 milligrams, 2 grams from liter. Regarding the chemical composition, this gives you the label, the designation to the mineral water. Bicarbonated, sulfated, chlorinated, sulfurous, sodium caustic, etc. So on. So if here you have the thresholds, the different thresholds, you consider to have the label of bicarbonate, sulfated, sodium, so on. And then you have also a very important point. When you talk about sulfur waters, you can have less than 500 milligrams, and but with this title of sulfur, you consider sulfurous water. So to finish, and in conclusion of my lecture, if we go to get a treatment or to have health benefits more than to treat a disease, I can go to a station, a thermal station, to promote my health. What do we have as a comprehensive therapy? Of course, we're talking about thermal medicine, and World Health Organization recognize this kind of practice, this type of practice, and calls it health resort medicine. So we're talking about procedures of diagnostic and treatment, not only treatment, also diagnostic. And we do not only treat, but we have health promotion. So we have health promotion by having better prevention, a better therapy, and a better rehabilitation. This is what you are doing in the field. Then the core elements of this complex therapy, thermal medicine, is the use of natural mineral waters, gases, and pellets. That's why we are here on a natural products conference, because we use, in fact, natural products. But we also use tap water. Now we're talking about complementary techniques, for instance, that we call hydrotherapy. When we use mineral waters, we call it crenotherapy. Creno comes from the Greek krenos, which means fountain, source. So we make the difference between crenotherapy, we use natural mineral water, and hydrotherapy, which use common water, tap water, plain water. So in spa therapy, in thermal, we can use, for instance, on collective practice, such as hydrokinesiotherapy, exercise on swimming pools, for instance. This is not crenotherapy, but could be thermal medicine by complementary techniques such as kinesiotherapy on pool, hydrokinesiotherapy, rehabilitation on pool. Okay, we use also climatic factors. This is very important. This is a natural variable, okay? So you can have similar waters, but in different climatic ambience, and you have different results. We have other therapeutic factors, namely sociologic, psychological. I gave a lot of importance to the interactive social interaction in therapy, and this is could be an excellent tool to improve our health. And of course, the contextual environmental factors. Now, we have also the methods or the modalities, baths, showers, drinking cures, inhalation, other techniques of respiratory treatments, immersion without or only to body parts, and the agents, the substance, the factors, CO2, sulfur gas, brine, carbonated, sulfated, etc. Then we can do underwater exercise, we can perform thermotherapy, we can perform hydrogalvanic baths. So physical medicine is also very important as a tool for this complex therapy. So as you see, to design a study on the effects on thermal therapy is not easy because we have lots of variables, and we need to be very careful to prevent bias and confounding factors. So the methodology to do research on medical hydrology, on thermal medicine, is very complex. Nevertheless, nowadays, I must tell you that in a recent bibliographic systematic review, I found thousands of studies of very good quality and more than 300 randomized control trials. And although it's very difficult to design blind controls, I say that I find some of them, and even double-blind studies are possible, but very difficult. So I thank you very much for your attention, and I am at your disposition to answer your questions if I can. Thank you very much.
Thank you very much for this excellent keynote, very clarifying. I learned a lot of thermal products. So now we pass to the questions. We have two questions. One from, it's written, is from Dr. Marcio Carocho. He says, thank you very much for the presentation. It was very broad and complete. I'm just curious about what you said about Água das Pedras. How could it be that the water that we drink rained over 1,000 years ago?
Thank you very much, Dr. Marcio Carocho. It's a very interesting question. In fact, we in thermal medicine, we hydrologists, doctors, we work a lot with the geologs. Professor Graça, hydrogeologists are our pharmaceutics in thermal. Not only the pharmaceutics, we count with you, but we need the scientists from geology to help us and to understand. And so it's very simple. Rain falls. It has the capability of penetrate into the soil, into the ground, and as a long, long way, going deeply on the rocks, having this way, and by some geologic occurrences, for instance, a geologic fracture, they are able to ascend and to come and to emerge naturally. Of course, we can do this artificially, and if we know that we have an aquifer, a pool of mineral water on the deep, we can also detect and then make a hole, make a perforation, and with that forage, we can reach the water. This must be very, very wise, very, very scientifically controlled and rigorous. Otherwise, we can have a natural disaster. And unfortunately, there are several of these disasters that kill that treasure, that preciosity of natural mineral water by bad practice on this kind of procedure. So again, the great majority of mineral waters have a long way during a long period of time, and this time allow again to have the water enriched with mineral salts and other contents such as biological, to have the product that, for instance, with Água das Pedras we got. And this is very interesting, of course, and nature is a precious gift, so we need to respect them. And I must confess my concern about the way sometimes we deal with water, mainly.
Okay,
and of course, climatic changes that are on the table now. These are due to the alterations, to the change of the water cycle. That's what it is. We have no more water. We don't lose water. We have the same amount of water in our planet for millions of years. Like Lavoisier law says, we don't lose water. We don't create water. Simply, the cycle has changed. And this is the problem we have, because it's changed quickly without the possibility to adapt ourselves. And our health is mainly found, like our life, our biological life, but also our economic life is based on water. Point. So pay attention for how you deal with water. So you need a lot of knowledge more, especially in medicine. Just a final thought. Doctors pass 6 years on medical schools in university, then 5, 6 years on specialized, and they don't talk about water. There is only one chair of medical hydrology in Portugal, which is mine. So it's incredible. It's incredible because water is, we are water. We are water. We are generated in water. All the biological processes we have are on water environment, and absorption, circulation, body functions are on water. And water is the mean to get better pharmacological effects and supplies. So this is a very important thought. And those in our Polytech Center are dedicated to natural products should in fact have this very important thought in mind. Thank you.
Okay, thank you. Just one more question, very brief, from Professor Maria Graça Campos. She wants to post a question and I'm going to pass her the word.
Thank you.
We are not listening, Professor Graça Campos, please. Okay.
Okay. Probably a challenger. I have a dream. I work with lots of oncologic patients. And one of my dreams is called for the cancers not so advanced to do the treatments in these thermal facilities. First, for the concept, and also because we can have a controlled ambience. So we could have a proper diet in an ambience that was friendly. And of course, we can have all these information that you have also. In all this research that you have already done, did you cross with something that we could in fact use this to put a project?
Oh, this is really the $1 million question. Really. Well, I'll try to be very short. It's not easy to be very short, but I'll try to put the problem. First, in fact, thermal therapy is a complex therapy. So we deal with nutrition, with exercise, and things that improve your health. Now, you can fight a disease by 2 ways. You can attack the pathogenic mechanisms. And if you don't know them, for instance, like COVID, what you do, you can enhance your body defense, your what I call salutogens, health gens.
Sorry, okay? My question was mixed. So
yes,
we have
of course.
chemotherapy, but
I think Professor Graça, everything in medicine, in my opinion, is mixed. Even when you give an antibiotic drug, you have the effect on the antibiotic on the microorganisms, but you have also the immunomodulation of of course. So everything is mixed. Exactly. Concerning cancer, we have, regarding mineral water, some mineral water show in lab some potential, some mitogenic potential. So we were much afraid of having cancers especially on the first period before remissions. We were afraid.
Okay, I see.
But then, we thought about the complex therapy, and now we enter the specific research for cancer. So nowadays, for instance, in France, there is a research line concerning several cancers. And we started by the rehabilitation of cancer disease. And we have now already some clues, let's say some signs that we have positive effects. And again, let's separate things. the pharmacy mineral water, and then the comprehensive therapy. You need to study both. You need to study the specific action of that specific composition for the specific cancer, and the immunomodulation that we know that some mineral waters can give to specify some response of antibody response, and then the comprehensive therapy. So I would say that I'm convinced that the next 10 years will bring interesting data on that research.
Perfect. Thank you.
I think.
So thank you very much for you both for the excellent keynotes. And now we'll pass for the oral communication section for the health category, and that which will be moderated by my colleague, Dr. Maria Inês Dias.
Thank you, Soraya. Good morning, everyone. I'm Inês Dias, and I will moderate the second session for the health issue of today. We will start with an invited oral communication followed by 6 oral presentations. Just a reminder for the participants of the Congress, you can leave your questions in the comment sections of the live streaming in YouTube, and you can also consult and download the posters on the Congress website. Without further ado, and because we are a little bit late, I would like to present our guest for the oral communication, Dr. Ana Novo Barros. Good morning, Dr. Barros. Hope everything is okay with you. Dr. Barros is an assistant professor with habilitation at the chemistry department of UTAD, and also the director from the Center for the Research and Technology of Agro-Environmental and Biological Science, named CITAB. Her area of expertise is the valorization of byproducts, taking into account the circular economy and industrial symbiosis, and her work has been recognized with the attribution of several prizes and awards. Congratulations, Dr. Barros. Today, the work that Dr. Barros will be presenting for us today is entitled Fighting Diabetic Foot Ulcers, Resorting to Winery Byproducts, Grape Steams Case. Dr. Barros, you have 8 to 10 minutes for your presentations, and I will pass it on to you. Thank you.
Thank you very much. Good morning, everyone. And before I start my communication, I just would like to tell you that I'm going to do this in the memory of my father that just died last week with a degenerative disease. And I'm really a believer that natural compounds can be so helpful for the prevention and for the treatment of this and the other diseases of our time. Now I'm going to share my communication. I'm sorry, but I'm having some problems. I think it's okay now. Can you please tell me if you can see it?
Yes, we can.
Please?
Yes, we
okay.
can.
Thank you so much. And now, starting with the presentation, and before everything, I would like to thank to the organizing committee the opportunity and the invitation to present a work that has been developed in the University of Trás-os-Montes and Alto Douro, in particular in the Center for the Research and Technology of Agro-Environmental and Biological Sciences, CITAB. The work that I'm going to present is entitled Fighting Diabetic Foot Ulcers, Resorting to Winery Byproducts, Grape Stems Case. This is the culmination of several years of work in the valorization of grape stems cultivars, giving them an added value. My presentation is divided in 5 topics: the introduction, the objectives, the material and methods, the results, the results in discussion, and the conclusions. Starting by the introduction, the antibiotic resistance is one of the biggest public health challenges of our time. Around the world, it is well established that about 2.8 million people get an antibiotic-resistant infection, and more than 35 million people die because of this. So our time with antibiotics is running out. Antibiotic resistance is a public health problem worldwide, having an impact in morbidity and/or mortality rates, especially in developing countries. According to the World Health Organization, in 2050, the antibiotic resistance will be the biggest cause of death worldwide, overcoming deaths from cancer, for example. One of the main causes of hospitalization are diabetic foot ulcer infections, mostly caused by Staphylococcus aureus, and due to the frequent use of antibiotics, Staphylococcus aureus strains have developed resistance to treatment. To combat the bacterial resistance, alternative antibiotics should be developed to overcome the effectiveness of the currently available. That's how it comes the grape stems case. Grape production is one of the main economic activities in the agri-food sector around the world. It is estimated that 52% of the production goes to the winery industry, whereas large quantities of byproducts are generated, namely organic residues, just like grape pomace, seeds, pulp, skins, grape leaves, grape stems, and wine leaves. Grape stems, which correspond to 25% of the total byproducts, is less characterized and evaluated of the all byproducts generated in the winery industry, although it represents up to 7% of the raw material processed. Primarily, the grapes are harvested, and before the vinification process, the stems are removed since their presence increases the astringency due to its richness in proanthocyanidins, affecting the wine's quality. So, usually it is discarded in open areas or used for composting, but it can cause some environmental problems and consequently it can compromise the sustainability and the competitiveness of this industry. So it is very important the adoption of new approaches, providing the application of these byproducts in the development of innovative and added value products that can be used not only in food, but also in cosmetic and pharmaceutical industries, since they are rich in bioactive compounds that present important biological properties for human health. These biological compounds are mainly flavonoids, stilbenes, phenolic acids, and condensed tannins. Due to the presence of these bioactive compounds, these matrix presents important biological properties such as anti-inflammatory, anti-diabetic, neuroprotective, anti-tumor activity. So it is very important to develop new products to combat antimicrobial resistance against not only against food diabetic ulcers, but also against other antimicrobial resistance. Thus, the objectives or main goals of this work are the identification and quantification of polyphenols present in grape stems from different varieties by spectrophotometric and chromatographic methodologies, the determination of antioxidant capacity of ethanolic extracts of grape stems, and the evaluation of antimicrobial activity not only from diabetic foot wound bacteria, but also against gastrointestinal bacteria from patients from the hospitalar center. To achieve these main goals, our sample was composed by 6 grape stems varieties, 4 red varieties, and 2 white varieties, and all the samples were collected in Quinta do Pinto in Alenquer. The grape stem extracts preparation was according with this procedure using food ethanol as solvent, once it is an environmental friendly solvent, and it is also compatible with food, pharmaceutical, and cosmetic industries. For the quantification of the phenolic composition, we have used the following methods for the determination of total phenols content, the complexation with aluminum chloride for flavonoids determination, and complexation with molybdate for the orthodiphenols content determination. The qualitative and quantitative analysis of phenolic compounds was done by HPLC, and for the identification of the phenolic compounds, it was taking into account the standards, the rotation times, order of elution, UV spectra, and literature data. For the antioxidant capacity determination, it has been applied the ABTS, DPPH, and FRAP methodologies. For the antimicrobial activity evaluation, and as I told you in the objectives, we have used gram-positive and gram-negative bacteria, not only from the foot diabetic ulcers, but also from the gastrointestinal tracts. So we have used the two different methodologies, the disc diffusion assay and the minimum inhibitory concentrations. We have used it as negative control, the MSO, and as positive control two antibiotics, gentamicin and ciprofloxacin. So as a result of our work, I can show you that about the phenolic content of the grape stems that we have studied, the total phenols and orthodiphenols presented the same behavior, and you can observe that Touriga Nacional is the variety that presents the highest amount for these two parameters, and they are significantly different from the others. And about the flavonoids content, although Fernão Pires is the variety that presents the highest value, it was not significantly different from Touriga Nacional and Tinta Roriz. About the identification of the phenolic compounds, we have identified in the all variety extracts with the difference of the anthocyanins that were just identified in the red varieties, 11 compounds that were grouped in phenolic acids, flavonoids, and stilbenes. Once the antioxidant activity is correlated with content of phenolic compounds, once again, Touriga Nacional presents the highest levels, and in all the methodologies used, the values are significantly different from the other varieties. About the antimicrobial activity of grape stem extracts and by the disc diffusion assay, concerning gram-positive bacteria, all the studied varieties revealed the inhibition of bacterial growth, and in some cases, this antimicrobial activity revealed to be highest than the antibiotics that didn't reveal any activity against two bacteria. In gram-negative bacteria, grape stem extracts only inhibited the growth of Pseudomonas aeruginosa. I can also tell you that, sorry, I'm passing this part, I can also tell you that in the concentrations tested, the extracts from Touriga Nacional and Syrah presented the best results affecting the specific growth rate of Staphylococcus aureus isolates and revealed a bacteriostatic effect. So as a conclusion of this work, we can say that grape stem is an effective matrix in the inhibition of gram-positive bacteria, especially for two of the bacteria studied. The grape stem extracts that presented antimicrobial activity revealed to be more effective than the antibiotics that were tested. And finally, we can say that grape stem can be reused for the cosmetic, pharmaceutical, and food industries. However, further studies are needed. To finish my presentation, I would like to thank the collaborators of this work, Professor Maria José Saavedra, Professor Eduardo, Dr. Irene Calvinhas, and Dr. Carla Dias. And sorry, but it has a problem. I don't know why. I would like just to show you some photos of our campus. I think that you can see it and invite you, all of you, to join us in UTAD when this pandemic situation finishes. Thank you very much.
Dr. Barros, thank you for your presentations. First of all, in the name of the Congress organization, we would like to show you our condolences for your father. It is a beautiful gesture that you show him tribute with your work, with an excellent work. So he would be very proud, of course. Dr. Barros, I would just like you to ask you two questions. Do you perform like a correlation study between the composition in phenolic compounds and the biological effects that you study? And do you believe there are other type of compounds present in the samples that you study that are responsible for the biological activities that you want to study?
Well, we are studying the grape stems in two different situations. We are studying the biological compounds about the phytochemical composition, and in that case, we have made the correlation also in the individual compounds. We have made the HPLC preparative, and we have seen that, for example, some white varieties presented biggest antibacterial activity than the red varieties. And we have seen that in some cases it was the presence of the vinifrin, for example, that was responsible for that activity. The other study that we are making, it's about the macromolecules that are present in grape stems, just like cellulose, hemicellulose, tannins, and so this is two different kind of studies that we are in course.
Thank you so much, Dr. Barros. I believe there is no more questions for you, but if someone has a question that want to pose to Dr. Barros, he can contact us and we will reach to Dr. Barros so she can answer you. Thank you again, Dr. Barros. We hope to see you next time. Good work. Thank you.
Thank you. Nice day. Good work. Nice.
So let's move on to our 6 oral presentations. We will begin with Ana Teresa Serra. Good morning, Anna. Anna will present to us the work entitled Unveiling the Protective Role of Natural Bioactive Compounds Towards Colorectal Cancer. Anna, I will pass to you. Thank you.
Thank you for the introduction. So, good morning. I would like to thank the scientific committee of this online conference for selecting our work for presentation in this session. Today, I will talk about the work that we have been developing at IBET, focus on the study of natural bioactive compounds in colorectal cancer. So it is well known that the Mediterranean diet is associated with a reduced risk of colorectal cancer, and this has been attributed to the presence of bioactive compounds, namely phytochemicals, that are reported to present anti-cancer effects. In this field, cruciferous vegetables, citrus fruits, and olive oil have been reported to contain glucosinolates, which are the main precursors of the more bioactive compounds isothiocyanates, phenolic compounds such as polymethoxylated flavones and hydroxytyrosol, which present anti-cancer effect in vitro and in vivo. However, the majority of these studies were performed on cell growth on monolayers or animal models that present several limitations. So the monolayer cultures present the limitation in mimicking the 3D tissue architecture, while animal models present ethical dilemmas and they did not mimic the human tumor microenvironment. So during the last years, our team has been working on the development of 3D cell models of colorectal cancer to elucidate the protective role of natural compounds derived from brassicas, citrus fruits, and olive oil towards this disease. So the 3D cell model was generating by culturing the HT29 cell line in in stirred culture systems using these spinner vessels. And using this system, we generate cell spheroids with different cell diameter and characteristics during culture time. In particular, the spheroids collected at day 7 of culture presented features of in vivo solid tumors, such as an apoptotic core, an hypoxia region, and also a stratified cell population and a structural complexity. As you can see here, the absence of epithelial cells in the spheroid outer ring and the presence of cancer stem cells in this spheroid outer ring. Additionally, this model presents a higher percentage of cancer stem cells when compared with the cell cultures. So stem cells have been identified in all types of tumors and have been highlighted to be responsible for tumor initiation, relapse, and chemo resistance. So they have been pointing to be a promising target for cancer treatment. So we use this model to evaluate the anti-cancer potential of natural extracts and bioactive compounds. So in the case of brassicas, our aim was to evaluate the effect of isothiocyanate-enriched extracts from watercress and broccoli in targeting cancer cell proliferation and stemness using this model. So the extracts were developed using supercritical fluid technology that showed to be effective in extracting these compounds. So watercress extracts present phenyl isothiocyanate as the main compound, and the broccoli extract presents sulforaphane, but also other isothiocyanates and fatty acid derivatives. So when we tested in our 3D cell model, both extracts inhibit cancer cell proliferation, induced cell cycle arrest and apoptosis, and modulated cancer stemness. Importantly, only watercress extract showed to reduce the cancer stem cell population. In the second case study, our aim was to evaluate the effect of polymethoxylated flavones isolated from orange peels in improving the antiproliferative of a chemotherapeutic drug. So the polymethoxylated flavones were isolated from orange peels in order to valorize this type of agro-food residue. In this work, the supercritical fluid technology was also used to extract this compound, and as you can see here, it was very selective in extracting polymethoxylated flavones. Senescentine, nobiletine, tangeritin, and scutelarine tetramethyl ether were the main compounds isolated from the orange peel extracts. So the results showed that the orange peel extract inhibited cancer cell proliferation and target cancer stemness in the 3D cell model. Important combination studies with the 5-fluorouracil demonstrated that orange peel extract, the mixture of the polymethoxylated flavones and also the compound alone improved the antiproliferative effect of the drug, as you can see here, the line presented in black. The highest synergistic effects were obtained between tangeritin and scutelarine tetramethyl ether and the 5-fluorouracil, and this could be related with chemical structure of these compounds. In the last case study, this study was focused on the evaluation of the anti-cancer potential of phenolic compounds from virgin olive oil. So the compound selected was hydroxytyrosol and its two main colonic metabolites, such as phenylacetic acid and hydroxyphenylpropionic acid. It is important to mention that these compounds were previously detected in human feces after a diet supplementation with a phenol-enriched olive oil, which means that they can reach the colon to exert their bioactive effect. we demonstrated that hydroxytyrosol was the most effective anti-proliferative compound in both monolayer culture and in the 3D cell model. And hydroxytyrosol was the only compound able to reduce the cancer stem cell population and inhibiting the colony forming unit capacity, which means that this compound impacts the self-renewal ability of the cancer cells. Overall, our results show that the 3D cell model is a promising tool to study the effect of food bioactives in colorectal cancer, and the phytochemical enriched extracts developed in our work, and also the isolated compounds, isothiocyanates, polymethoxylated flavones, and hydroxytyrosol present antiproliferative effects and modulate cancer stemness in this advanced model of colorectal cancer. It is important to mention that the knowledge generated in this type of in vitro studies provides relevant information towards the design of human intervention studies for cancer treatment. Finally, I would like to acknowledge to all the people and collaborators involved in this work from IBET, IPO and CIAL, our partners from the food industry, and also the funding received from IBET Export Project, Innova for Health, FCT, Cost Action, and Program of FITEC. And thank you for your attention.
Thank you, Anna Serra. Thank you for your wonderful presentation. Since we are a little bit running out of time, I would like to tell all of you from this first session to wait until the end of the session so we can gather all the questions, and then we will make up the time that we have. Okay, so thank you, Anna. Again, let's move on to our second presentation that will be done by Carlos Gomes. Good morning, Carlos. Carlos will present the work entitled Anti-Aging and Neuroprotective Activity of Thymus carnosus, aqueous and hydroethanolic extracts. Carlos, let's move on to you. Thank you.
Just a minute. Something is wrong.
Okay, we can see it now. Yeah, we can share.
Thank you. Sorry. So, good morning all. Sorry for this delay. I will be presenting my work, which I would like to thank for the opportunity to present. It's about the anti-aging and neuroprotective effects of aqueous and hydroethanolic extract of Thymus carnosus. Thymus carnosus is an endemic plant from the Iberian Peninsula. It can be found in some populations within Portugal and Spain, south coast, and also in Portugal, southwest coast. And due to its restricted location, it has been listed as an endangered species, as it can only be found in natural parks and natural reserves. Apart from this, it can also be found in some collections in UTAD's Botanical Garden that were the location where we harvested our plant for this study. So for the methodologies, as usual for aromatic and medicinal plants, we selected the aerial parts that were grounded and lyophilized and grounded, and were then used for the extraction procedure. We performed two extract procedures. One, an aqueous extract mimicking the human consumption as either an infusion, teas, or as a condiment in food through the boiling process. And also an exhaustive hydroethanolic extraction method using 80% ethanol that we were aiming to extract 100% of the phytochemicals present in our plant material. So both our extracts were analyzed by HPLC, both DAD and mass spectrometry, resulting in chromatograms as we see here in this image. Apart from normal compounds that we can find in aromatic plants within the thyme genus, as glycosidic derivatives of some flavonoids as luteolin or apigenin, The main core of our extract was composed of phenolic acids, namely newer compounds as salvinoleic acid A new isomer of this acid we named salvinoleic isomer A, that was the phenolic acid present in higher contents in our extracts, also commonly found rosmarinic acid, and salvinoleic acid K with high content within these plant extracts. We verified that extract-dependent concentration was obtained due to the effect of the ethanol in the hydroethanolic extract, and also the use of ethanol allowed us to extract another class of compounds, two pentacyclic triterpenoids, ursolic acid and oleanolic acid. Ursolic acid was the compound present in higher amounts in our extract, in both of them, in the hydroethanolic extract, sorry. So regarding this plant, due to previous studies, we already know that it had antioxidant activity, performed well against several human carcinogenic cell lines, and showing it anti-proliferative activity, and also some anti-inflammatory activity. And our study was to evaluate its effects in anti-aging and neuroprotective activity, since other time species, as for example Thymus pelagioides, had shown great results in this type of bioactivities. So we choose to evaluate three enzymes: acetylcholinesterase, tyrosinase, and elastase. Acetylcholinesterase and tyrosinase were chosen for the neuroprotective activity due to the involvement in metabolic pathways related to neurodegenerative diseases as Parkinson and Alzheimer, and also tyrosinase and this time elastase for their effects in anti-aging activity due to the relation to metabolic process within the skin. What we observed was that both extracts greatly inhibited acetylcholinesterase activity, higher inhibition for the hydroethanolic extract, most likely due to the higher content of phytochemical phenolic acids, flavonoids, and the pentacyclic triterpenoids, and selective inhibition of elastase and tyrosinase was observed. Only aqueous extract inhibited tyrosinase, and only the hydroethanolic extract inhibited elastase. In order to correlate these findings to our phytochemical composition, we selected three of the major compounds that were available as commercial standards: rosmarinic acid, ursolic acid, and oleanolic acid, that we see that in acetylcholinesterase inhibition, as expected, three of the three compounds presented inhibition concerning this enzyme, also correlating to the results we obtained for the extract, since the hydroethanolic extract presented higher inhibition, and only the hydroethanolic extract had ursolic and oleanolic acid. Regarding tyrosinase, none of the compounds at the tested concentrations presented inhibition, once again correlating to the results, since if these compounds would present some inhibition, then hydroethanolic extract would have higher content and therefore a higher inhibition. And all the standards inhibited elastase activity, once again showing correlation with our results, since most likely the concentration within aqueous extract of rosmarinic acid is not enough to produce an inhibition, and since we only have the triterpenoids in the hydroethanolic acid, only there the inhibition was observed. And so, as conclusions, we showed that our extracts have a potential for neuroprotective activity, and extract composition-dependent inhibition of both elastase and tyrosinase was observed and correlated with the phytochemical composition. And once again, we highlighted the potential of Thymus carnosus as a source of functional foods and also as a source of nutraceutical compounds. I would like to thank FCT for the funding to both the research centers and my PhD grants, to the laboratories where I conduct my research, and of course, my supervisors. Thank you.
Thank you, Carlos. Thank you for your presentation. As I was saying, let's leave the questions for the end of the session. So I will invite you to be with us until the end of your session. Just a quick note, not for you, but for everyone, for the participants. I'm sorry, Carlos. If someone has problems in entering the Congress website, please use another navigator, especially for the Mac users. We were told that some of you have problems in entering the page website. So let's move on for the third presentation of this session with Carlos Sharaishi. Good morning. Good morning, Carlos. Yes, good morning. Carlos will present the work entitled Anti-Inflammatory Evaluation of a Leap of Natural Compounds Identified in Mushrooms Using In-silico Studies Against COX Enzymes. So, Carlos, let's move on to you. Thank you.
Thank you very much. Hello, it's a pleasure. My name is Carlos. It's a pleasure presentation my research in this Congress, intitulated Anti-Inflammatory Evaluation of a Library of Natural Compounds Identified in Mushrooms Using In-silico Studies Against COX Enzymes. Cyclooxygenase, COX-1, is a protein present in physiological COX-1 and inflammatory COX-2 process. COX-1 participates in the metabolism of arachidonic acid, acting first in oxidation and later on reduction, converting arachidonic acid in prostaglandin G2. Protein COX-1 and 2 are the target of inhibition for non-steroidal anti-inflammatory drugs for the treatment of diseases. NSAIDs. There are some examples of NSAIDs: mefenic acid, aspirin, ibuprofen, and etoricoxib. So NSAIDs is using for treatment of diseases: asthma, cardiovascular, colorectal cancer, diabetes, neurodegenerative diseases, osteoarthritis, and rheumatoid arthritis. So, used for a long time cause adverse effects, according for European Medicines Agency, such as gastrointestinal, renal, and cardiovascular problems. So, need alternative to compounds. Therefore, it is necessary to identify new molecular entities that act by inhibition COX-1 and 2, or according to Wooler, there is a cost of approximately 1.8 billion to introduce a new drug on the market. Therefore, computational tools are important in the development of drugs to assist in the screening of compounds. This research increased and prepared the compound library from 115 to 190 mushroom compounds and identified the best one with COX inhibitory potential. Performed the individual screen study, pharmacokinetic, docking, and molecular dynamics. This present methodology is identify proteins in protein data bank database and evaluate their quality, identify molecules with medicinal potential from mushroom available in the literature, design and or preparate ligands and proteins in PDBQT format, evaluation of the best software, Autodoc tools or Autodoc Vina, automation of the docking process with Mola software, using Swiss admin server for obtaining pharmacokinetic data, and Dismount software to perform molecular dynamics. My materials is my computer, Spin, and let's go to the results. The expansion of the library 3.0 was a success. The library is composed to 190, and the family's compound consists of 43% terpenes, 40 steroids, and other compounds, isoflavones, flavones, catechols, phenols, quinones, and others. So, the LMW library 3.0 library has a satisfactory pharmacokinetic profile according to Swiss admin server with evaluates the pharmacokinetic parameters or admit absorption, distribution, metabolism, excretion, and toxicity. Vina was identified as the best software for COX simulation, performed a satisfactory forecast compared to experimental results through constant inhibition. Virtual screening was performed. The factor compounds for molecular docking. The compounds origin A from the Neonotophanes nambu mushroom present inhibition constant 152.4 nanomolar, and clavulactone C from the Psittacibia clavipes mushroom had a highlight inhibition against COX-2 of 52.3 nanomolar, and compound of simalonic B is have interaction with the two proteins, COX-1 and 2, provenient from Vomitopsis officinalis. So, pharmacokinetic prediction, the factory compounds is have a good drug likeness profile according to Swiss admin server. The dynamic simulation for clavulactone C had the best key value, satisfactory pharmacokinetic profile, and was verified via molecular dynamics is stability within protein. The molecule does not diffuse from the protein in over time 10 nanoseconds. Conclusion, in general, terpenes were highlighted against the enzymes COX-1 and 2 from the docking studies performed using AutoDock Vina software. Origin A terpene produced the lowest predicted key value against COX-1 and clavulactone C with COX-2. Clavulactone C showed stability within the COX-2 through molecular dynamic simulation for 10 nanoseconds. When both COX enzymes were considered, oxymalonic B provides the best profile for dual COX inhibition, COX-1 and 2. So the three highlighted compounds maybe have anti-inflammatory potential, having as mechanism of action the inhibition of COX-1 and or 2. However, experimental verification must be performed. Thank you very much.
Thank you, Carlos, for your nice presentation, quick presentation. Thank you 2 times. So we will gather the questions until the end of the session. So let's keep you with us, and we will talk in a minute, okay? Thank you. Let's move on to the presentation by Clara Grosso. Good morning, Clara. Good morning, she's with us already. Clara will talk with us about silver brain, neuroprotective potential of seaweeds, subcritical water extract. So Clara, let's move on to you. Thank you.
Good morning. Thank you very much for the invitation to present our results in the project Silver Brain. This is a project, a collaborative project between researchers from Requinte in ITZEP and Faculty of Science and Technology, and also researchers from IBET. In this project, we try to take advantage of the biggest and the richest number of seaweeds distributed worldwide, which are rich in several metabolites, and we try to think about what about search for new neuroprotective drugs that could be very useful and beneficial for people with ages over 65 years old and to contribute to active aging. However, working with seaweeds, it's not easy because we have to take into account several associated problems. First, we have to think that waters where they are produced in aquaculture or they are naturally occurring can contain contaminants, environmental contaminants such as pharmaceuticals, pesticides, and heavy metals. And also some seaweeds are very rich in iodine, and their contents can be very high and can disrupt thyroid and contribute to the development of thyroid disorders. Moreover, preparing extracts there's also another issue that we must to take into account, that are the solvent residues. And therefore, in order to overcome this last problem, we opted by a green technology to extract the bioactive compounds, which was the supercritical water extraction. We performed the extraction in a temperature gradient mode in order to obtain with the same raw material several fractions from room temperature to 250 degrees. The experimental outline were grouped in three main parts. First, we tried to assess the presence or the absence of several environmental contaminants, which included pharmaceuticals, pesticides, and also their transformation products and metabolites by LCMS, and also some one heavy metal and iodine content by ECP-MS. Concerning the extracts, the bioactivities and the chemical composition, we target our investigations for right now for spectrophotometric methods to assess the chemical composition, but we hope that in future we can go further for LCMS analysis, and some in vitro bioactivities targeting the oxidative stress and also enzymes involved in several neurodegenerative and psychiatric diseases such as Alzheimer's, Parkinson's, and depression. Concerning the assessment of the presence or absence of contaminants, we were glad to see that all the extracts were free of pharmaceuticals and pesticides. However, they have a problem of high amounts of iodine and arsenic. As you can see by the graphics, in the first steps at 19 degrees and 140 degrees, there are a high amount of iodine and arsenic that are leached. I would like to call your attention that as long as the extraction progress, the amounts are even less and less and less. But in the case of the arsenic content, I would like to call your attention that in the first steps, the kind of arsenic that is leached is the inorganic one, which is the more toxic one. So we cannot use the first and second step of the extraction for further assays. And then at 119 degrees, in the case of Fucus vesiculosus, we can say that there's a little bit more arsenic that are leached, but it is the organic fraction, which is less harmful. So taking all these results into account, we decided to go further for the bioactivities assessment with just the step 3 and step 4 of the extraction. For both seaweeds, the Fucus vesiculosus, that is a brown seaweed, and Cardon that is a green seaweed. Concerning the bioactivities, in the case of the antioxidant activity, you can see that the lowest IC50 value, which means the concentration that inhibits the radicals or the antioxidant activity in 50%. For the superoxide radical and for the nitric oxide radical, we can see that the more active fractions are the ones that were obtained in the last step of the extraction at 250 degrees. Therefore, we decided to go into more activities to see if they are beneficial for neuroprotective activities, and then we tested only the fractions 3 and 4 of these two seaweeds in several enzyme assays. As you can see, for the acetylcholinesterase inhibition and butyrylcholinesterase inhibition, that are enzymes involved in Alzheimer's disease, only the extract of Cardon obtained at 250 degrees were active. And the same for the other enzymes. The fourth step were always the most active one. So we decided to go look and see what kind of chemical composition was these bioactivities were due to which kind of chemical compounds. And as you can see, the phenolic compounds that are the most widespread compounds in seaweed extracts, you can see that they are decreasing from step 1 to step 4 in the case of fucus vesiculosus, but there are a little bit increase in the Cardon tomentoso case. But we have to take in account that there are several interferences in the phalanxio test, so we need to see these results with some caution. And the same for the total flavonoid content and fluorotannin content that is decreasing along the extraction. So we decided to see in the literature what kind of compounds could contribute to the bioactivities at 250 degrees. And we came across with the Maillard reaction that is well described in the subcritical water extraction processes, because it takes high temperature, and it results from the reaction of reducing sugars and amino acids that we could see that are increasing from step 1 to step 4 in our extractions. As you can see, the colorless intermediates Maillard reaction products, the yellow
Clara,
yes.
2 minutes, okay?
Yes. The yellow ones and the brown ones are always increasing along the extraction, and they can contribute to the bioactivities because they are already reported as antioxidant compounds. Therefore, we can conclude that the subcritical water extraction is a very versatile technique to obtain several fractions from the same raw material. They can eliminate from step 1 to step 4 high amounts of heavy metals and iodine, and at the same time we can obtain extracts with good activity in terms of antioxidant and neuroprotective effects. And these bioactivities can be a combination of a low amount of phenolic compounds and a high amount of Maillard reaction products. Thank you very much. I would like to thank the organizing committee for the opportunity to present results, and to all the financial institutions and all the researchers that contributed to these results. Thank you very much.
Thank you again, Clara, for your talk, for your wonderful presentation. Let's move on to Katja Kramberger presentation. We will ask you the questions in the final session. Okay, Clara. So Katja Kramberger will present the work entitled Helichrysum italicum infusion stimulates energy expenditure and fat oxidation after acute ingestion in humans, a pilot study. Katja, hello, good morning. Let's move on to you. Clara, please stop sharing the...
Sorry.
Yeah, thank you,
Yes.
thank you. So Katja, let's move on to you. Thank you.
Thank you for the introduction. Just a second. Okay. Yes, I will present to you a pilot study with Helichrysum italicum that we conducted on healthy male volunteers. The study was performed by our research group at the Department of Nutritional Counseling within the Faculty of Health Sciences at the University of Primorska. We are located at the coastal region of Slovenia, and our research is mainly focused on Mediterranean plants and functional foods in general. But lately, our focus is mainly on Helichrysum, which is also a topic of my PhD work. Many of you are probably familiar with this plant. It is characteristic of Mediterranean region. It can be recognized by its yellow fate-resistant inflorescences, which are rich in bioactive secondary metabolites. And these bioactive compounds are synthesized as a result of adaptation to this demanding environment. Apart from volatile terpenes, which are mainly present in the essential oil, the plant is also rich in phenolic compounds, which are rather polar compounds, and also well-known antioxidants. Due to its rich biochemical profile, Helichrysum italicum possesses many promising pharmacological activities. This plant has been traditionally used for respiratory infections, digestive problems, and for inflammatory skin conditions. But it has been also investigated scientifically, both in vitro as in vivo. But mainly on single isolated compounds. Therefore, we wanted to investigate whole plant water extracts, so-called infusions, which are often used in traditional medicine. Our aim was to explore acute effects of Helichrysum italicum infusions on resting metabolic rate and possible substrate differences in substrate oxidation in healthy male subjects. We also performed additional in vitro experiments to further investigate the mechanism of action of Helichrysum infusion. An infusion was prepared by immersing 1 gram of Helichrysum in boiling water for 10 minutes, and then chemical composition was determined as well. And for the pilot study, 11 male adults were divided into 2 groups. And assigned to either bioactive beverage or placebo beverage, which was just a hot water. And then to the other beverage after 7 days of the washout period. Metabolic rate and substrate oxidation were measured by indirect calorimetry at pre-ingestion. This was at the baseline, and after 30 and 120 minutes post-ingestion. And for the gene expression analysis, RNA was isolated from infusion-treated and control hepatocytes, and we used HepG2 cell line for this purpose. We identified various chemical compound classes, among which caproalkynic acids were the most abundant, and followed by pyrrones and flavanols. Using calorimetry measurements, we found that a single ingestion of Helichrysum infusion caused a slight but a significant increase in energy expenditure in healthy male subjects compared to the placebo beverage, as well as a decrease in respiratory quotient after 30 minutes and still after 120 minutes post-ingestion. A similar trend was also observed for fat oxidation, which was also increased compared to the placebo. However, no significant change was observed for the blood pressure over time, except for the diastolic blood pressure at the 30 minutes post-ingestion. The same effect was also observed, confirmed in vitro experiments regarding fat oxidation, where we found an upregulation of fatty acid genes, genes responsible for fatty acid oxidation, such as CPT1 and CPT2, and also a downregulation of ACC gene, which is involved in fatty acid synthesis. However, no significant change was observed for DGAT, which occurs as the terminal in the terminal step of triglyceride synthesis. With the present study, we have gained some insight into the activity of Helichrysum italicum in humans, which was very scarce before. We have shown that Helichrysum italicum infusion stimulates both resting energy expenditure and fat oxidation in normal weight men, which was also confirmed in vitro on molecular level. This poses a possibility that consumption of Helichrysum italicum may have a beneficial effect on an individual's ability to maintain lower body fat levels. However, further studies under pathophysiological conditions and in long-term use are necessary to draw more solid conclusions. I thank you for your attention.
Thank you so much, Katja. Thank you for your wonderful presentation. Let's leave again as your colleagues the questions until the end of the session and move on for the last presentation of this session by Teresa Braz. Hello, Teresa, good morning. Teresa will talk with us about Chitosan-based films loaded with cynaropicrin enriched extract from Cynara cardunculus anti-inflammatory potential. So Teresa, let's move on to you. Thank you.
So good morning. As was already said, my presentation is about the Chitosan-based films loaded with cynaropicrin enriched extracts from Cynara cardunculus and the assessment of its anti-inflammatory potential. Okay. But first, let me talk you about cis-quaterpine lactones group that for sure are familiar for most of you and comprises over 5,000 known compounds. Cis-quaterpine lactones are considered one of the most prevalent and biologically significant group of secondary metabolites with described biological activities such as anti-tumoral, antimicrobial, antimalarial, anti-inflammatory activities, among others. And for that, they are considered a valuable resource in terms of health applications. Besides having a good amount of biomass production that can range from 7.8 to 20 tons dry weight per hectare, Cynara cardunculus is also a great source of cis-quaterpine lactone, and for that was selected to be used as a case studies in this project. As observed by Ramos et al. During the lipophilic fraction characterization of Cynara cardunculus leaves extracts, cis-quaterpine lactones may have a content near to 95 grams per kilogram of dry weight. And from the ones identified, cynaropicrin was considered the one in major quantities with almost 87 grams per kilogram of dry weight. Several studies had focused on cynaropicrin high biological potential, where anti-inflammatory potential is of particular interest when thinking in terms of wound dressing applications. Since extensive inflammation plays a major role in the disruption of normal healing cascade, presenting a significant psychological, physical, and financial burden for patients. When compared to systemic delivery, localized drug delivery systems may reduce undesired side effects such as toxicity and suboptimal delivery. Besides, they provide spatiotemporal control over the drug dosage, directly at the wound site and also protects the drug from metabolic deactivation and maintains the drug concentration at the desired level. Nevertheless, an appropriate and immediate coverage of the wound area with adequate dressing is essential for wound protection in order to accelerate wound healing. And in a constant need to improve patients' lives and decrease ambulatory costs, in the last 2 decades, efforts have been made toward the design of new wound dressings with the incorporation of bioactive materials such as chitosan, regarded one of the most promising materials for wound dressing due to its unique properties. That can be improved by incorporation of bioactive compounds such as cineropicrin, that in combination with its anti-inflammatory potential may lead to an enhanced wound healing. And from that, the global aim of this work was the design and production of cineropicrin from Cynarda cardunculus and enriched leaves extracts and its application on a chitosan matrix as a wound dressing for anti-inflammatory drug delivery. And what concerns to the film development, they were developed by the solvent evaporation method, where chitosan membranes were loaded with different extract concentration and obtaining dense and thermal stable membranes. In what concerns to the mechanical properties, namely the puncture stress, the films produced presented a lower mechanical resistance when compared to the control one, what was mainly attributed to the presence of hydrophobic compounds such as cineropicrin, and also to the molecular interactions between the extract and chitosan, leading to the weakness on the polymer chain aggregation forces. Nevertheless, the produced films were still able to be applied as a wound dressing since the human skin tensile strength is located between 5 and 30 megapascals. The swelling effect was also evaluated, and also a decrease on the film swelling capacity was observed with increasing extract concentration, what was assumed to be correlated to the decrease of the polymer hydrophilicity, leading to a lower swelling ability. This lower swelling ability indicates that developed films are suitable to be used on light exudate wounds. According to the international standard guidelines for biological evaluation of medical devices, a cell viability reduction higher than 30% is considered to present cytotoxic effects, and the analysis of the results for the cell viability assay using the BJ cell line indicated that the films with 1 and 5% extract were not considered cytotoxic, with toxicity highly correlated to the cineropicrin extract content. With IL-6 as one of the key pro-inflammatory factors produced by dermal fibroblasts in response to certain stimuli, normal human skin fibroblasts were stimulated using liposaccharides from E. coli and exposed to film extracts in order to understand the film anti-inflammatory potential. And the results revealed that a slightly decrease with the lower extract content was observed, and the strongest effect was observed for the 5% extract with a reduction of 86% and 83% in the IL-6 expression by skin fibroblasts comparatively to the stimulated cells and also to the ones exposed to the commercial anti-inflammatory piroxicam. When comparing the IL-6 effect of the extract and the cineropicrin standard, a more potent anti-inflammatory was observed for the extract comparatively to the cineropicrin. These results disclose the potential to the extract to be used as a wound dressing for chronic wounds. To conclude, at the end of this work, it was possible to conclude that to observe that a decrease of the volumetric swelling capacity was observed with the extract loading. What could be advantages for wound dressings where an excessive fluid absorption is not desired? Also, a decrease on the tensile strength with the extract loading was observed, although still inside of the skin parameters. The film loaded with 10% extract presented cytotoxic effect to the BJ fibroblast cell line, what was correlated to the cineropicrin content. On what concerns to the IL-6 levels on the LPS-stimulated skin fibroblasts, The film with chitosan and chitosan loaded with 1% presented the same effect obtained with commercial anti-inflammatory pyroxicam. The film, on the other side, the film with 5% extract presented a significant reduction on the IL-6 levels, showing its potential for chronic wound management. To finish, I would like to thank to the research and to the research institutions, to the funding institution, the Faculdade de Ciência e Tecnologia, and to funding projects, Biotech Sinara, Medcinara Biotech, and also to all the research team, and to you for your attention. Thank you.
Thank you, Teresa. Thank you for your wonderful presentation and fast presentation. Thank you for that. Let's move on now for a little session of questions. I have here 3 questions and no more. We do not have time for more also. The first question is for Ana Serra. So Ana, are you listening to me? I don't know if Ana is here.
Yes.
Yes, okay. Anna, the question that I have here for you is, are there already in the market some natural-based medicines for the purposes of your study? Did you were able to compare results in terms of efficacy with your natural products?
In the case of hydroxytyrosol, for instance, there are already companies that are selling the compound isolated for food and for cosmetic applications. Concerning the use of this type of compounds for cancer, this is not regulated well, and yes. There are the claims that are already reported in the EFSA, are more related with cardiovascular diseases and metabolic diseases. So for cancer, it is quite difficult to,
yes, of course. So regulation is the next step for your study, and we hope that all of this is towards that path. Thank you again, Teresa. Another question that I have for Carlos Shiraishi. Hello, Carlos, can you listen to me? It's a question from Marcio Carocho. He says that, thank you for your interesting presentation. I saw you use Autodoc Vina for the docking studies. Any specific reason for using that one? And did you try any other docking program to compare your results?
I compared my results with the experimental values disponível in Pit Protein Data Bank. I'm using protein inhibition protein and realized the cross docking. I'm validation my simulation in this mode.
Okay. Thank you again, Carlos. The last question is for Katja Kramberger.
Yes.
Katja. Yes, hello, Katja. The question that I have for you is, did you ever thought on using other type of preparation extracts besides infusion? Do you think that the infusion extract is the best one to obtain the amount of bioactive compounds that you need to see the health effects that you want?
Our primary goal was to use such preparation that is the most suitable for the consumption in humans. We looked up for the preparation that are mainly used in traditional medicine, and then the most common ones are infusions or decoctions. namely some water-based preparations that are easily to prepare, easily prepared. Yes, that's why we used this one. But also in our previous study, where we investigated chemical composition of Helichrysum in general, Helichrysum italicum in general, we also used ethanolic extracts and methanolic extracts. These are somehow richer in, of course, richer in phenolic compounds,
phenolic,
but are not so appropriate for ingestion. Ethanolic, yes, but methanolic definitely.
No, it's a question of safety and the final purpose of your extract. So thank you, Katja, for your answer. Thank you all again for your wonderful presentations. Thank you again for Dr. Ana Barros for the invited lecture that she gave us to us. And let's move on to the pitch session and the last session of this morning. So I will now pass the word to my colleague Filipe Reis that will moderate the next session. Thank you all and good morning.
Thank you, Inês. Good morning, everyone. We will continue with our participants' communication session. Who will present us the main results of their research in developing and applying natural products in the health area. Let's start with the pitch sessions. I want to remind the audience that you can follow the conference's live streaming on YouTube's IPB channel. You can follow the communications and you can send your own questions through the chat. And after selection, I will present those questions to our speakers. You can also download the posters available online. You will find on the conference website 3 links corresponding to the 3 areas covered in this Congress: health, food, and cosmetics. Regarding the pitch sessions, I inform our speakers that they have 3 minutes to make their presentations. There will be no interruption between the presentations, and only at the end we will open a space for questions. To not cause major delays for this pitch session, we will allow you to ask only 1 or 2 questions. The remaining questions could be sent by email, okay? So let's move on. Ana Sofia Pereira de Freitas will start the session. Ana, you can start your presentation. You have the floor.
So, good morning, everyone. My name is Ana. I'm a PhD student from the University of Minho, Portugal, and I will present a part of my work regarding the antibacterial and antioxidant synergism between propolis and gentamicin or honey. Since the first case of bacterial infection with resistance in 2008, antimicrobial resistance has become an emergent problem, causing a prediction of 10 million deaths globally by 2050. The overuse of antibiotics, their extensive use in farming practices, their patients' misuse, along with the lack of new antibiotics, are contributing factors to this problem. So there is a need for strategies to overcome the microbial resistance and development of new antimicrobial drugs. Interest in natural products has been increasing due to their spectrum of bioactivities that can be used for the development of new drugs to retard the outbreak of bacterial resistance, decrease bacterial resistance impacts. So propolis is a natural product produced by bees from several plant materials and mixed with their salivary enzymes. So after the determination of the minimum inhibitory concentrations for both gentamicin and the ethanol extracts of propolis, by the agar dilution method, several mixtures of submic concentrations of both agents were tested in order to find synergism. So as you can see in this table, with a submix of 25 micrograms per milliliter of propolis, it was possible to reduce to 25 times less the amount of gentamicin initially needed to inhibit the growth of bacillus strains. Also for Staphylococcus aureus methicillin sensitive and resistant strains and for E. coli, it was possible a reduction of 70% of the amount of gentamicin initially needed to inhibit bacterial growth. Regarding propolis and honey synergism, the higher the percentage of honey in the mixture, the lower the antioxidant capacity. However, with the exception for Bacillus cereus, all the mixtures of submix concentrations of propolis and honey showed strong antibacterial activity than each individual sample, with particular interest for these strains, where it was found a huge reduction of the amount of both agents. So this outcome is significant considering the worldwide problem of antibiotic resistance and the need to innovate in antibiotic therapy, in particular if solutions from natural sources, such as propolis and honey, are envisaged. So thank you for your attention.
Thank you, Anna. Thank you so much. Now we are moving on to André Folgado. You may present your poster, your pitch session, please.
Yes, so you are hearing me? Okay,
Yeah.
So thank you. Good morning, my name is André Folgado, and today I'm going to talk to you about air-root cultures of Cinera cardunculus as a valuable source of hydroxycinnamic acid compounds. So, as you already heard, Cardon is a plant widely studied regarding their secondary metabolites. The upper parts of the plant already showed some important secondary metabolites, although this is true for the above ground part of the plant, the underground part of the plant still remain pretty much unstudied. To address this question and this issue, we used air-root cultures. So these air-root cultures are the result of Agrobacterium rhizogenes infection. These are characterized by a high growth rate and also high branching. They are important because they produce the same secondary metabolites as the source plant, and they are characterized by a high genetic stability, which give this a high potential for biotechnological applications. So for the induction of the air roots from Cardon, we infected hypocotyls from Cardon, and after we had the air roots, we perform air root methanolic extract, and we analyze this extract by HPLC-MS, and we detect several compounds, although this mixture showed low complexity. So the peaks are very well resolved. So 3 of them were confirmed by standard, and the rest of the peaks were confirmed by comparison with the literature. All of these compounds belong to the hydroxycinnamic acid group, and all of them are chlorogenic acid derivatives. Then we analyzed these cultures in their growth curve, and we analyze them in terms of total phenolic content that increases along the growth curve of the air root cultures, and the same pattern is true for the antioxidant activity, reaching its highest value at the 35th day of the growth curve. So we use this time point to produce a concentrated extract that was further used to test antiproliferative activity on human colorectal cell lines, HT29 cells, that presented an EC50 at 1.16 milligrams per milliliter. The same concentration showed no effect on cells that mimic the intestinal epithelium cells, human cells. So with this, I thank you, and I finish. Thank you for your attention.
Thank you, André. Now, Bruna Mara Machado Ribeiro will present her pitch. Bruna?
Bom dia, pessoal.
Bom dia. Bruna, you can start.
Good morning. Okay?
Okay.
My name is Bruna Mara Machado Ribeiro. I'm from Brazil, work as scientific research. My line is neuropharmacology, neuroinflammation, neuroprotection, and neuroimmune activation. My studies detect inflammatory effect of N3 polysaccharide fatty acids on virus infect neurons. The viral mimetic poly-IC is important to study the consequences of viral infection to the development of neuropsychiatric disorders. Based on the premise of omega-3 presents anti-inflammatory effect, was investigate the involvement of NF-kappa B pathway in the effects of omega-3 in hippocampal neurons exposed to the polycell virus. Poly-IC is a synthetic immunostimulation virus. Poly-IC induced by microglial activation. Poly-IC increased to levels in us, NF-kappa B, P5, P65, ELIC, nitrites, and hippocampal neuronal cells. Poly-IC causes deficits in neuronal hippocampal mechanism. Omega-3 prevent neuroinflammation alterations induzidas por poly-IC. Omega-3 prevent the deficits de BDNF. Omega-3 increases DCX immune expression. Omega-3 presents effect anti-inflammatory. Poly-C induced microglial activation. Poly-C induced increased the levels of NF-kappa B P15, NF-kappa B P65, omega-3 effect anti-neuroinflammatory, effects neuroprotectives omega-3 in neuronal cells, MTT increased, neuroprotective effects omega-3 lipid peroxidation, omega-3 suppresses the expression of P15, 65, NF-kappa B subunit, neuroprotective effects of omega-3 in NOS, neuroprotective effects omega-3 in P15, NF-kappa B in NOS, co-immune stent, in primary hippocampal neurons, neuroprotective effect omega-3 in P65, NF-kappa B, protein expressed in primary hippocampal neurons, changeling, neuroprotective effects omega-3 in interleucina 6 levels in primary hippocampal neurotendons, poly-C, neuroprotective effect of omega-3 in nitrite levels in primary hippocampal neurons, omega-3 prevent the deficits BDNF in cells, omega-3 increases the CGX immunoexpression. Thank you very much. Good morning.
Thank you, Bruna. Now I call Catarina Faria Silva to make her presentation. Good morning, Catarina.
I will share my here. Okay, can you hear me?
Yeah,
okay.
Perfectly.
So good morning, I'm Catarina Silva from Faculty of Pharmacy of the University of Lisbon. And today I will tell you about the extraction of alpha-tomatine from green tomatoes industrial waste to treat skin inflammatory diseases. Starting with green tomatoes. During the tomatoes harvest, many of them are still green and they are thrown back to the fields, being an industrial waste. Furthermore, it is known that green tomatoes are rich in glycoalkaloids, namely alpha-tomatine. And this glycoalkaloid was already studied in its pure form by several research groups, and they found out that it possessed several activities, namely anti-inflammatory, fungicide, anti-carcinogenic activity. So what we are proposing here is to establish a sustainable and circular economy where we are going to take the industrial waste of green tomatoes, extract the alpha tomatine through an environmentally friendly method, the subcritical water, and then we are going to incorporate the alpha tomatine in a topical formulation to treat the skin inflammatory diseases. The extraction, this thermodynamical process is a very important part of this work because we want to assure that the process is as green and sustainable as possible. And subcritical water basically uses only water, high pressure, and the tomatoes. We had to develop these methods, we tested several conditions, and in the end we obtained an extract. To have a comparison, we also performed the extraction with conventional organic solvents with a method that is already described in the literature. However, the extract has all the components of the tomatoes. So in order to quantify the alpha tomatine, we need to do an additional purification step. So we perform solid phase extraction, and what we found out is that when using the subcritical water extraction, we obtain 10 times more alpha tomatine than with conventional organic solvents. So what we are going to do next now is to do a characterization of the full composition of the extract, and thank you for your attention.
Thank you, Catarina. Now we have Ala Haddad.
Hello.
Hello, good morning. You can make your presentation.
Yeah. Hello. Hello, my name is Hela, and today I'm going to present a project titled by Optimization of New Anti-Inflammatory Methodologies to Evaluate Natural Product Activity as Inflammation Inhibitors. I'm going to start with the introduction and objectives. Inflammation is a defense mechanism that is designed to eradicate microbes or irritants to protect living tissue from infections, injuries, and to potentiate tissue repair. The objective is to exploit Zingiber officinalis and Vironia dioica as promising anti-inflammatory agents. Methodology. We are going to use the hydroethanolic extract to evaluate the anti-inflammatory activity using mouse macrophage, cytotoxic activity using 4 tumoral cell lines and 1 normal cell lines, and the cell cycle and apoptosis using flow cytometry. Result and discussion. The results are still preliminary and needs to be repeated. The anti-inflammatory activity. We are going to measure the EC50 value, which is correspond to the extract concentration achieving 50% of the inhibition of NO production. The 2 samples and their combination present a really interesting value, and the BD present a very low value, which is even lower than the positive control. Cytotoxic activity. We are going to measure the GE50 value, which is correspond to the concentration of extract that inhibited 50% of cell growth. For Z officinalis, the 4 cell lines present a low GE50, a lower GE50 than the normal cell line, and the NCE present the lowest one. For BD, all the value of GE was lower than 6.25, so we are going to repeat this test with the lower concentration. The cell cycle analysis shows that the Z officinalis had the same distribution, cell distribution as the blind test, and the B dioica has a really interesting increase in G0 and G1 phases, so it does interfere with the cell cycle. The apoptosis analysis shows that the BD have no apoptotic effect, and that the ginger, which caused a really interesting decreased percentage of dead cells, have an apoptotic effect, which is confirmed to the literature. Conclusions. Given the well-known side effects of the commonly used synthetic anti-inflammatory agents, this preliminary study contributes to the discovery of natural agents as real alternatives and with less or no side effects. Vironia dioica revealed the highest potential with the highest anti-inflammatory activity and the higher effect on the cell cycle distribution. The chemical characterization in terms of phenolic compound profile is ongoing, and that's really interesting parameters, so we can know the relation between phenolic compound composition and the biologic activity. Thanks for your interest.
Thank you, Ala. Finally, I invite Nayara Fernandez to present her work. Nayara, good morning. Is Nayara there? Yeah. We are not listening to you. You have your microphone.
I'm really sorry. I'm already sharing my screen?
Yeah.
Okay, perfect.
Okay.
Thank you. Sorry for the delay.
Don't
Okay,
worry.
so I'm Nayara Fernandez. I work in Nutraceuticals and Bioactive Process Technology Lab at IBET. And today I would like to show you a little bit about the industrial waste stream valorization that we are doing here at IBET. The competences in our food and health division includes the recovery of high added value compounds, chemical and bioactivity characterization, and finally, if needed, we also do some formulation for the addition of these ingredients in final products. We are used to work with different natural matrices, but today I will present 3 case studies that are based on wineries and fisheries waste streams. In this first case study, I would like to show you several terpene-based natural dipotectic systems were synthesized with the aim of recovering astaxanthin from brown shell residues. So those systems were synthesized and physicochemically characterized, and they're applied for the extraction of the mentioned carotenoid. The results show that the results were comparable to the ones obtained with conventional methodologies. And also they were applied to another matrices such as microalgae and other seafood. Also, this terpene-based system and the extract obtained showed to have good potential for anti-cancer and antimicrobial activity. This second work that I would like to present to you, we were able to extract phenolic and anthocyanin compounds from winery waste streams. We were able to enhance the yield of the extraction, applying a microwave pretreatment prior to the solid liquid extraction. And then those extracts were evaluated for their potential for the possible potential in cosmetic applications. They saw high antioxidant activity, both in chemical and cell-based assays, and also several enzymatic assays showed that they had some effect in skin aging. Finally, I would like to show you this cotton-like extractor, a fiber that was synthesized here by supercritical drying of gel that was formed by alginate and chitosan, both of them very abundant polymers in marine environments. So this material showed to be non-cytotoxic and also showed to have a strong antimicrobial effect. Moreover, it stimulates the migration of skin cells. So overall, it was seen like a possible candidate to use in wound healing applications. And that's all. Thank you for your attention. Thank you for the conference committee for allowing me doing this presentation. And please, as it was very fast, if you have any doubt or comment, please contact me by email and we can discuss further. Thank you.
Thank you so much, Nayara. We have here one question. First of all, thank you all that present your pitch. All were very clear. And I have here a question sent by Marcio, one of our participants. He thanks Catarina for her presentation. And he thinks it's a very nice idea. So Catarina, Marcio is asking, why did you choose the subcritical water to perform your work? Why? And if you choose another one or tested another one?
Well, like I said, we compared with organic solvents that is already used in describing the literature, but the subcritical water allows us to have what I show much more tomatine, alpha tomatine, and it's a green process, only uses green solvents. So it goes in line with the sustainable idea that we want to have in our final product.
Okay, thank you, Catarina. Since we are running late, I guess we will move on. I remind all the participants that you can send your questions via email for us, and then the speakers can answer you, okay? But since we already exceeded the scheduled time, I guess we will move on, okay? So let's move on to the morning's last presentation in the health area, the Keynote 3. As for the previous sessions, do not forget that you can follow the conference's live streaming on the IPB channel on YouTube, and you use chat to place your questions. I remind you that you should send your questions during the presentation and do not save them to the end, as we may not receive them on time, and therefore we will not present those questions to the speaker. So Keynote 3 will be presented by Dr. Adelaar Bracht from Maringá State University, who I am pleased to present next. Dr. Adelaar holds a PhD in Natural Sciences from the University of Munich and is currently a senior professor of biochemistry at the State University of Maringá. He has been doing research work since 1980, focusing on liver metabolism, including oxidative stress. I welcome Dr. Adelaar and invite him to present his keynote entitled The Adjuvant-Induced Arthritis Model and its Utility for Investigating Anti-Inflammatory and Antioxidant Effects. Dr. Adelaar, welcome, and please, you have the floor.
Thank you. You are hearing me?
Yes, perfectly.
Okay. I will begin by sharing the screen. Well, good morning to all. Firstly, I wish to thank the organizing committee of this nice meeting for inviting me to deliver this speech, and say that it is an honor and it is a real pleasure to be here. As the title reveals, the title of this speech, I will be emphasizing and trying to convince you that of the importance of in vivo approaches for evaluating antioxidant and anti-inflammatory activities. I am the speaker, but in fact, we are a team at the State University of Maringá, which is situated exactly on the tropic Capricorn in southern Brazil. Actually, I am a retired senior investigator, and these are the names of my associates, which are engaged in research, teaching, and supervision. And of course, the lab work is being done or has been done mostly by our students. The names of most of them will be mentioned.
So sorry, Dr. Adelaar. I'm sorry, yeah, but we are not seeing your presentation. You did not share the screen. We are seeing you.
Oh, sorry. Is it okay now?
No, we are seeing you.
No, not yet. If you can go and share screen.
Let's see. Presentation, shared screen. And now?
Not yet.
Not yet.
Okay, I guess it's now. Start working. Okay.
I'm sorry for that. I'm sorry.
Don't worry. Okay, okay.
Okay. So as I said, I will try to convince you that the adjuvant-induced model is important for measuring anti-inflammatory and antioxidant effects in vivo. As I said, these are my associates and the students. I will eventually mention several of them as I will present our results, some of our results. Let me change this. So. Well, anti-inflammatory and antioxidant effects are generally the first phenomena that are, oh yeah. I'm still there now.
It happens.
No?
No, your presentation just go away.
I clicked something that I shouldn't have clicked. Well, let's start again.
Okay.
I don't know why it's not working.
Professor, if you wish, you can send us your presentation by email and we can post it for you.
Let me try again.
Professor Adler, can you hear me?
Yes, I can.
Okay, I think you've got it now. Okay,
yeah.
It's we can see it.
Is my screen there?
Yeah.
Yes, you may continue. Yes.
Okay. So let's move to the next. So there are many in vitro systems that allow a preliminary evaluation of these properties. In general, however, these characterizations are relatively uncertain because they are artificial conditions, which mostly have little or no resemblance to the in vivo conditions.
especially the effective doses and concentrations are frequently uncertain. These are the main reasons why in vivo models are in fact needed. These models represent an intermediate step that precedes the final goal to be reached, the clinical trial. The adjuvant-induced arthritic rats present several favorable features for being used as an in vivo model. First of all, several inflammatory, oxidative, and metabolic indicators are modified. Modifications are essential. This is because it is extremely difficult to improve conditions that are already close to normal. The modifications in these rats, these arthritic rats, are not restricted to the joints, but they usually become systemic and affect a lot of organs and tissues. The modifications in these rats, furthermore, are very similar to the modifications that occur in rheumatoid arthritis. This is important because this generates a translation factor. So you can translate eventually these results to rheumatoid arthritis patients. Because in patients, in the actual patients, limited interventions are possible. You can obtain, for example, blood samples, or eventually samples from other tissues. In the rat, however, all organs can be analyzed. So you will have a parameter diversification, which increases certainly the importance of the results you are obtaining. In the rat, arthritis can be induced, for example, by injecting the so-called adjuvant, which may consist of HIV inactivated Mycobacterium tuberculosis, for example. If this adjuvant is injected, for example, in the left paw, an edema develops quite rapidly, which is evidenced by this paw volume increase that is observed here. Later on, the non-injected paw, in this case the right hind paw, also develops these lesions. And in time, the disease becomes systemic. It extends to the whole body, so that practically all organs, or at least most important organs, are in fact affected by the disease. This produces many changes that can be measured. These changes should be sensitive to anti-inflammatories or antioxidant agents. Here we see what happens in the liver, for example. This work was published a few years ago, in which we analyzed the changes in the liver cells. This work specifically, but the scheme bases on several modifications that were detected by preceding work. As you can see, circulating cytokines induce several transformations, which include, for example, increased glycolysis, because glucokinase is increased, decreased glucose 6-phosphatase, which is in line with decreased gluconeogenesis, decreases in the NADPH-NAD ratio, which is connected with the decrease in GSSG ratio. And all this leads to several modifications, which include increase in reactive oxygen species, which means increase in protein oxidation, increase in lipid peroxidation, and so on. There is also an increase in nitric oxide synthase, decrease in antioxidant enzymes, and so on. All these parameters can be measured. And potentially, antioxidants, natural antioxidants, should affect these parameters. That is an important fact that I would like to emphasize here. These effects are compartmentalized. You see here 3 compartments. We have distinguished these compartments: the cytosol, peroxisomes, and mitochondria. The effects may be different in these 3 compartments. Ideally, one should not analyze the whole cell contents, but it would be more appropriate to analyze different compartments, although this is linked to hard work and is rarely done, in fact. So this gives us a general picture of what happens in the liver. You see, all these parameters can be measured or should be measured when testing and the antioxidant effect of a given compound. Similar things happen in the cardiomyocyte. We have published this paper a few years ago too. The effects are equally compartmentalized and may even be different depending on the parameter. For example, thiols are increased in the endoplasmic reticulum, but decreased in the endoplasmic reticulum, but increased in the mitochondria. The extent of the effects may be different, but in general, the same parameters that are modified in the liver are also modified in the heart. A similar picture we have in the brain. This is a neuron, and we show in this picture the main modifications that may occur when the rat becomes arthritic. Now, arthritis induces a series of modifications in the brain, as you can see, which are similar to those that occur in the liver and in the heart. But here we have also measured the transmembrane potential of the mitochondria, which is increased, a fact that certainly contributes to the increased production of reactive oxygen species, which lead to lipid peroxidation, protein oxidation, modifications in the level of GSH, which is a decrease in this case, and modifications in the nitric oxide concentration. The mechanisms, the basic mechanisms, are always the same. There is an increase also in xanthine oxidase, which is very pronounced in the brain, and which is a parameter that can be measured when you are investigating the effects of natural compounds on the oxidative status of these rats. Well, these are the main changes that occur and that can be explored when you are measuring the effects of natural compounds. We have done this with a series of natural products, purified or not.
We have done this, we have investigated the action of several extracts with generally convincing results. I am showing here, before I show some of our results, another picture about the liver, which shows modifications in metabolism. Arthritis modifies metabolism in several ways. As I already said, glycolysis is increased, gluconeogenesis is decreased, respiration is increased. This is generally coupled to an increase also in the oxidation of fatty acids, as shown by this work that we have published more recently. The mechanism is always the same. By the way, adipose tissue is equally affected. We have not investigated adipose tissue, but it is equally affected. So you have an increase in the oxidation of fatty acids, which leads in increase in production of CO2. In consequence, you have an increase in the respiratory chain, which in the brain at least also reflects an increase in the mitochondrial membrane potential. Changes in the NAD ratios, and so on. These are also parameters that can be measured, and it would be highly interesting to know if natural antioxidants and anti-inflammatories really affect these metabolic parameters and not only oxidative stress indicators. Of course, there is an increase in reactive oxygen species and so on. But things are correlated. There is an intense cross-talking. So it would be interesting to investigate metabolic parameters as well. Next, I will show you, I have not so much time, of course. I have wasted some part of my time in trying to present this speech because I am an old-fashioned man. So I would like to show you results that were obtained with two different agents. An extract, which is an extract of Ilex paraguayensis, which in English is normally called yerba mate, using this Spanish word, in Portuguese, erva mate, or other designations here in Portuguese and Spanish, which is an herb which is consumed in southern Brazil, in Argentina, Paraguay, and Uruguay. In this way, these girls are drinking what is in fact a hot water aqueous extract of this herb. The herb is dried, still green, and it is used to prepare this beverage here. It is very rich in methylxanthines, which is caffeine and theobromine, for example. It is highly stimulating, but it also has other effects. In our study, we used aqueous extracts mimicking the way by which the herb is traditionally consumed. For example, these are measurements in the plasma. These are plasma parameters. I have selected some parameters. I'm not showing the entire work, which has been published in Food and Function 2 years ago. This work was done in collaboration with the Mountain Research Center, which determined the content in phenolic compounds of the extracts that we use. You see here, it is important to use more than one dose of the drug. It is important to use appropriate controls, which is the normal rat, which was also treated with 2 doses of the extract. The arthritic rat presents increased plasma protein carbonyls, which measures the lesion to proteins, which a parameter measures lesion to proteins. There is also an increase. Sim?
Não faz mal. Estive a falar com a professora e por acaso estava a falar com ela porque tive outros problemas hoje de manhã.
I'm so sorry.
So, arthritic rats present increased myeloperoxidase levels, this is plasma, it decreases the thiol levels in the plasma, and decreases FRAP reactivity in the plasma, which is a measure, can be a measure of antioxidant capacity. As you can see, treatment attenuates these effects to some extent, sometimes more, sometimes less. The effects are not always that pronounced, but there is a clear effect, attenuating effect. This is an inflammatory indicator. This is a oxidative stress indicator, an oxidative stress indicator, and also an oxidative stress indicator. The effects are different. The extents are different. This cannot be predicted from in vitro experiments. Similar results were obtained when the liver was investigated, as you can see here. Protein carbonyls are increased in the liver as well as in the brain. RAS levels are increased in the liver as well as in the brain, and treatment with the extract decreases these levels. It does not really normalize these levels, but this certainly and apparently increasing the dose will not increase the effect, but there is an effect, a decreasing effect, normalizing tendency when arthritic rats are treated with these extracts. GSH, glutathione, reduced glutathione, is not really much increased than normal rats are treated with the extract, but they are decreased by arthritis, and there is a partial recovery, as you can see. But the effect is much more pronounced in the GSH-GSSG ratio, which is actually a redox ratio, and which is much more important. This occurs in both liver and brain. So what I want to conclude. I want to conclude about this few results that I showed to you, that you can indeed, in the arthritic rat, measure and detect antioxidant and anti-inflammatory effects, which are predicted by in vitro measurements. But the in vitro measurements are not able to predict the extent and the doses that are required to produce these effects. And there are also other parameters that are not affected. They should be affected, but they in fact are not affected. I have not enough space to show all these results, but I can assure you many parameters are simply not affected in spite of the intense antioxidant activity of the extract. Ideally, ideally, one should always use purified compounds. In the case of the yerba mate, Ilex paraguayensis, there are no purified compounds available, or most of them are not available. Chlorogenic acids, you generally cannot get great amounts of these compounds. But there are other compounds that are available, readily commercially available, as for example, resveratrol, which, as you all know, is quite abundant in grapes, for example, and in many other fruits or products of plant origin. Well, resveratrol is a panacea. Many effects have been attributed to it. What interests us is the anti-inflammatory and antioxidant effects. And we have done recently a work in which we have changed our mode of doing experiments with natural products. Instead of using a single dose or two different doses, a low dose and a high dose, we have used various doses, several doses. We have done a multiple dose investigation for several reasons. Multiple dose investigation, although the committees, the ethic committees do not like, because you must use many rats, many animals for your study, I think it is important because it can bring to light many different effects and particularities of the actions of various compounds. This work, which was recently published, shows that resveratrol inhibits, in fact, the production of reactive oxygen species in a concentration-dependent manner in isolated mitochondria. These mitochondria were isolated from the brain, where these are isolated mitochondria, so that all factors have been eliminated, all factors that might be increased production of reactive oxygen species in vivo have been eliminated. Even so, this is important, in my opinion, to investigate the action of natural products in isolated mitochondria because you may be surprised, and we have been surprised in several ways, in several occasions, because sometimes the compound may act as a pro-oxidant, in fact, and not as an antioxidant. Well, here are some results that I have picked to demonstrate the action of multiple doses of resveratrol, a clear concentration-dependent effect on the myeloperoxidase activity in plasma. As you can see, in arthritic rats, it decreases the activity. No clear effect in control rats, but resveratrol was not able to modify the diminished plasma albumin concentration. And the effect in control rats is a mess, difficult to interpret. But
there was a decrease in the increased xanthine oxidase activity. But here again, the effect in control rats was a mess. No defined effect, a declining tendency here, but this is difficult to interpret. A almost normalization of the effects, of the negative effects of arthritis in catalase. Dose dependent, of course. And I would like to emphasize before I end my presentation, the effects on plasma thiol concentration, which is illustrative of the reason why we should do multiple dose experiments. As you can see, arthritic rats present low concentrations of plasma thiols. This is consistent with oxidative stress. And this is mainly due to the low levels of albumin, of plasma albumin, as plasma albumin is the source of most thiol, reduces thiol groups. Well, the various doses of resveratrol had no effect on the thiol group concentration in plasma. On the opposite, they had an increasing effect on the plasma thiol content in healthy rats. This is antioxidant effect. The plasma albumin concentration was not changing, in fact, but its ability to have antioxidant effect has increased when resveratrol was given in a concentration-dependent manner, but there is a maximum here and a decline afterwards. So actually, this shows that resveratrol has two kinds of effects: antioxidant effect at low doses, and later on, a pro-oxidant effect. This appears quite clearly in healthy rats, but nothing happened in arthritic rats. But here another factor appears, another factor intervenes. In arthritic animals and patients, there are higher levels of glyoxal and methylglyoxal. These compounds tend to promote resveratrol-induced oxidation, even at low doses. So actually, the interaction of resveratrol with glyoxal and methylglyoxal is actually impairs the possible antioxidant effect of resveratrol on plasma albumin. In conclusion, you can conclude that all these things can in fact not be predicted by in vitro experiments, actually not even in cell systems. You have to use animals, unfortunately, to discover or to see experimentally all these different effects. So let me conclude. So in our experience, the adjuvant-induced arthritic rat is an appropriate and convenient model for investigating anti-inflammatory, antioxidant effects. Advantages are similarity to the human disease, a fair number of parameters can be measured, several and important organs are affected, high reproducibility, pro-oxidant actions can eventually be detected. For the future, multiple dose experiments are recommendable for increasing both significance and validity. Effects on the metabolic parameters changed by inflammation and arthritis may produce new insights into the mechanisms underlying the disease.
thank you for your attention. I'm sorry for my lack of training in this kind of presentation. At the beginning.
So much.
At the beginning.
It happens all the time. So thank you, Professor Adelaar. I don't know, I guess our audience is very shy and they are not questioning so much.
Thanks, God.
No, but thank you for your interesting and clear presentation. And thank you for meeting the allocated time, even with those problems at the beginning. Okay, I think we are not receiving anything. So I just have one or two curiosities. They are not questions. We saw in your presentation, of course, and we know that you tested not only yerba mate or resveratrol. So when you tested some of the natural compounds that you choose, did you test them, all of them in their free form, like this aqueous extract, or did you isolate some compounds, or you have only results for steel beans like resveratrol? You tested mostly extracts in their free form?
Up to now, most of our works, our studies have been done with extracts, but always characterized in terms of their contents.
Ah, okay.
Characterization, as I don't know if I mentioned it, is generally done at the Mountain Research Center. It's a collaboration that we have with this highly productive group. But we have also done several studies with isolated compounds. I gave you this example with resveratrol, and we have several other studies in which we use the purified compounds.
Okay. So
the trend, of course, is to use purified compounds, so you can really know what compound exactly is doing what. This is very, very important, I think. But sometimes it is difficult to obtain the main products that exist in extract.
Yeah, of course. So gathering all the data obtained in your work regarding the natural compounds that have shown promising effects in this inflammatory process inherent to the rheumatoid arthritis disease, you cannot establish yet a relationship between the obtained results and, for example, the compounds present in these extracts and infer about the most promising classes of compounds, right? We don't have those answers.
Yes, you have a group of compounds, but you can't say with certainty what exactly compound is responsible for what, as I emphasized.
Okay, of course.
You can translate the doses. Look at Ilex paraguayensis, there is a lot of consumption. There are people that drink these infusions during the whole day. So they consume a lot of these compounds. You can make translations because they are formulas to the human case, to the human situation, I mean. You can make translations using certain formulas, of course. But it would be highly desirable if all these compounds were available in pure form. But you need sufficient amounts. Because in vivo experiments consume much greater amounts of specific drugs. In vitro, you have very, very small volumes and you can test at will. You can repeat tests and so on. This is a disadvantage of in vivo experiments.
Of course.
But you can use very small animals. Instead of rat, you can also use mice, which are much smaller and require much smaller doses.
Okay, thank you. Okay, I guess the participants have hungry. They want to have lunch. So I think I will end the morning session that had as a theme the application of natural products in the health area. I thank all the speakers who presented us with their communications, and I also thank the entire audience for having accompanied us and for your participation. Any question or doubt, contact us through the links available on the Congress website, email, and social networks. Before I leave, I have just one message for André Folgado, one of our speakers in this morning's speech session. Since there were problems during your presentation, André, we were unable to view your slides. So if you want, you can make your presentation at the end of the day, at the end of the last pitch session, okay? You can contact us for any further information. So thank you, Professor Aguilar. Thank you all the speakers in this morning session. So I declare the session closed. We restart at 2:30 p.m. With a session of natural products applied to the food sector. Have a good lunch, everyone. See you in the afternoon at 2:30, okay? Bye-bye.
Thank you, goodbye.